Good afternoon. I'd like to welcome everyone to the second session of this symposium. My name is Sabrina Vasconcelos Caram, and I'm a biological science undergraduate student at the Federal University of Espírito Santo.
Hello everyone, my name is João Victor Nunes. I'm also a biological science undergraduate student at the State University of Feira de Santana, Bahia, Brazil. And we are members of the chapter Iniciação Botânica, a group of young botanists affiliated to the Brazilian Botanical Society. And 4 Brazilian universities from 3 Brazilian states are involved in this project, whose aim to introduce biology students and botany subjects and promote and support scientific events. This is the first event that the chapter is hosting this year, and I would like to invite all of you to follow us on social media because we have exciting news and events upcoming. Our Instagram is at Iniciação Botânica.
We're going to be the mediators of the session about Gao ecology and interactions. Now I'd like to invite Dr. Rosi Isaías from Federal University of Minas Gerais and Dr. Denis Oliveira from Federal University of Uberlândia to say some words.
Rosie, excuse me, your microphone.
Sorry. Good afternoon, all. It's my pleasure to have you here. Yesterday I enjoyed a lot, and I hope you have also enjoyed our meeting. And today we will have 6 fantastic talks. And in the name of the organizing committee, I welcome you all, and I will pass the words to Denis Oliveira to say something to the audience.
Hello everyone. One more great day for our symposium, Biology of Goals. I would like to thank you, the speakers, for taking our invitation. I hope you enjoy. It's a great event proposed by Neotropical Gao Group. Thank you.
Thanks for the words, Rosie and Denis. Now we will begin the session. Today we're gonna have 3 presentations. The first one is with Christian Villagra. Christian currently works at the Instituto de Entomologia, Universidade Metropolitana de Ciências de la Educación. Christian does research in entomology, ecology, and evolutionary biology. Their current project is characterizing the ecological niche of native cockroaches in Chilean biodiversity hotspot site and plant insect association. The second speaker is Dr. René Carneiro. René is a biologist graduated from the Federal University of Minas Gerais with master's and doctorate degrees in plant biology by the same institution. He studies structural and metabolic changes in plants due to the development of galls induced by insects. Dr. Carneiro is particularly interested in studying the natural history of the association of galling insects and their host plants, and also plant cell biology in the context of plant phenotype manipulation by galling insects. The last speaker is Bering Otieno. Bering has a Bachelor of Science in Agriculture from University of Nairobi, Kenya, a Master of Science in Agricultural Entomology from the same university, and a PhD in Zoology from La Trobe University, Australia. Perry Otieno currently works as a research scientist, entomologist, and assistant regional director at Kenya Forestry Research Institute. Her research experience spans most aspects of pest management, including biological control of crop and forest pests, pesticide efficacy, testing and application, host plant resistance, and general integrated pest management. Welcome.
Thank you.
Now we can begin with Bearing. Bearing is the first. Sorry.
Thank you. So I'll share screen.
Yes, you can share your screen.
Does that come? Did it come?
No, I can't see.
Can't see the window. Maybe cancel and start again. Oh, nothing seems to be happening.
You click in below.
Sorry?
In the name share. Below in the name share. Shares.
Open the what?
There's a button called share.
Yes, I see the button and I go to the application, but I can't find the window to share. And the share thing isn't active.
Did you click on share screen?
It say, there's an error message. Let me read it. Screen sharing was cancelled. Make sure you click or something. Are you able to enable me? Looks like something is stopping it from sharing. It can't come on. I can't seem to share it. Sorry about this. The share thing isn't active anymore.
You click in the. Do you click in the screen box? After clicking in sharing, you must click in the screen box. There appears in the screen.
Share at the base. The share screen at the base. Sorry about this. It's taking me too long to do it. It's not unable to share.
Can you send me the PowerPoint and I will
Sorry?
share? Can you send me the PowerPoint?
Yes, let me send it to you. How should I send it? Sorry?
Wait a sec.
Hi, Sabrina. Should I? Sorry?
The Gmail I put in the chat. You can see?
Sorry?
I put in the chat. You can see?
On the private chat.
On the private chat?
Yes.
Let me see. So in the chat? Okay. Mm-hmm. It's asking whether I have issues. I think this is taking longer than anticipated. Sorry, this is taking long.
Okay, so we can change the order of the presentations. I'm gonna put René Carneiro now, and then you go after. Okay, Bering? I'm gonna.
Hi. Sorry, everybody. It seems that Bering has a problem sharing her screen. So we're going to invert the order a little bit, and then we can put my presentation on while she sends us her PowerPoint presentation so we can go on, okay? I made a video of my presentation, so I won't take you too long. And please, Sabrina or João, could you share my screen, please? It's shared over there.
Hello, everyone. Good afternoon. Thank you for being here today. I am René Carneiro, and I will be talking about the curious case of Nototriosa species associated to Obsidian species in Brazil. I am professor at the Federal University of Goiás in Goiânia, and also part of the gall group research on eutrophical galls. This is my email address. If you need me, don't hesitate to contact me. Well, and why do I think the case of Nototriosa galls are so curious? Well, I'm going to tell you a story. Once upon a time, I had a master's to do. So while doing field trips, I ran into this species here, Psidium myrtoides, Myrtaceae, and I found out that this species was actually a superhost of galling herbivores. So you have here bud galls, a tiny leaf gall, and two other more globoid leaf galls. But as I started working with this species and collecting galls, I noticed that these three ones here were not very frequent. In fact, they were quite rare. So I started working with these two that are actually the same. They just range color from red until green, and is induced by Nototriosa species. So, if you were here yesterday, you already know a little bit about the story behind the history of these galls induced by Nototriosa. As Dr. Buchart said, the first records for Neotriosa was of this species, Neotriosa tavaresi, described by Crawford in 1925, based on the material collected by Priest Tavares in Bahia, Brazil. And here is a picture of that material. You can see the galls here. They are similar. And also the opened galls also look very similar. So when I sent the material of these galls to Daniel, he was suspicious that could be the same species or a closely related species. As you can see here in the title of my dissertation, the name is Neotriosa tavaresi, as to confer. Well, however, when we looked into the literature for the records of Neotriosa tavaresi in Brazil, we found that this report by Butignol and Pedrosa Macedo in South Brazil, where it was associated with Psidium catleanum. So the question came to our minds, could it be that Neotriosa tavaresi associated to 3 different host species? So we started to dig deeper on the taxonomy of those insects, and that led us to publish this paper, Biology and Systematics of Gall-Inducing Triosids associated with Psidium species in Brazil. And here we erect a new genus, Nototriosa, which is part of the Triosidae, and Neotriosa tavaresi is transferred to Nototriosa as a new combination, so its name now is Nototriosa tavaresi. And the 2 new species are described, Nototriosa catleani, here you can see the wing and the nymph, and Nototriosa myrtoides, the wing and the nymph. As they were very similar, I was very intrigued by that, and I decided to investigate those galls, which were very similar, during my PhD. But the question here is, why would that be interesting? Why study similar galls? Because we have here the case of closely related insect species associated to a single plant genus, both Psidium species. So here you have the galls of Nototriosa catleani on Psidium catleanum and the galls of Nototriosa myrtoides on Psidium myrtoides. And as you can see, the shapes are similar, the color range is similar, so we had to look for structural and biochemical peculiarities in these very similar gall morphotypes. We were basically trying to test the validity of the extended phenotype concept. That is, if these galls are truly independent and peculiar entities, at some level they should be different, and we wanted to discover where was that difference. And we started by looking into the development of those galls, and as you can see here in this cross section, we have the galls of Nototriosa myrtoides and the galls of Nototriosa catleani. And once again, the anatomy showed us that the phenomena related to the morphogenesis also regarded great similarities, as you can see here by the tissue layers, and also the emergencies growing to cover the insects. So we wanted to know more, and we started looking into gall functioning. And as we looked into the cells, we were amazed because look at these marvelous and beautifully active cells near the vascular bundles here in Nototriosa cattleani, here in Nototriosa myrtoides galls. Cells with large nucleus and conspicuous nucleoli, rough endoplasmic reticulum, mitochondria very developed, and the same patterns were observed here. You can see the sieve element tubes of the phloem and the parenchyma cells around it, denoting with many organelles denoting these nutritive capacity. So we described in these galls, actually they have nutritive cells in the vascular and perivascular parenchyma. So that was pretty cool. And as we looked further into the type of metabolites that were accumulating these galls, we've seen that they had starch, for instance, also near the vascular bundles, the presence of proteins, and we found that there was the activity of enzymes related to the metabolism of carbohydrates also around the vascular bundles. Here you can see glucose 6-phosphatase and invertase, and they basically make sugars available for the insects to suck from the vascular bundles. But there was not actually a novelty, because it had been reported before by Oliveira and Isaías on Pseudophacopteron galls on Aspidosperma. As you can see here, they also accumulate carbohydrates and proteins, for instance, and they also did have gradients of metabolites. But here it was very interesting to see that, to confirm the occurrence of such gradients, and to see that they were slightly different in different galls. And to conclude our analysis on these very similar galls, we started to look into the cell wall composition, and we did some immunocytochemical assays to see some cell wall components. As you can see here, the labeling of galactans by these monoclonal antibody LM5 that shows us that in the outer cortex and the medium cortex and toward the inner cortex, we have the gradual increase of labeling. So we also found gradients on the labeling of these cell wall components, and they are basically related to tissue functioning, such as increased rigidity, as in the case of galactans, but also porosity, flexibility, and so on. So what we found is that the cell wall composition was variable and different among the two galls, which led to different functional profiles of the cell and tissue layers in the gall, and also enabled the cells to expand differently. So we had different types of cell expansion within these galls during gall development, which means gall ontogenesis. So when we look at the processes of gall formation, we can truly understand these galls as true extended phenotypes of their inducers, even though they are morphologically similar. So when we came to this point, the question arises, so what is next? Well, one of these days, as I became a professor here at the Federal University of Goiás, I was doing my job, and then I received a photo of this plant. and that caught my attention because it was very similar to the goals I had studied before. So we started to investigate this plant here, which is, we found out to be another Psidium species, Psidium larotiano, here in the Cerrado, in the state of Goiás. And moreover, as I was doing a field trip on the Atlantic forest in São Paulo, I also found these galls, which were strikingly similar to the galls I had studied before. This is probably also a Psidium species. We couldn't identify it to the species level. So here we go again. We start to research on those galls once more. And here you can see some results of the monography of Ivonete Fernandes. We are preparing a manuscript on that, and also another few details about these galls. This is the galls of Psidium larotianum. As you can see, the processes are somewhat similar to what occurred during the induction of the other galls, but it has some peculiarities probably linked to the stressful environment of the Cerrado. But the development is quite similar, and we were able to report some cellular responses that were either similar or a few ones that were different. So this is in preparation and coming out soon. And the interesting thing here is when we look at the distribution of these plants on the map of Brazil, we can see that here is Nototriosa cattleyiana in Psidium cattleyanum. Here is the new species from Cunha in São Paulo. Here is Nototriosa myrtoides in Psidium myrtoides in near Belo Horizonte, Minas Gerais, and this is the report by Padre Tavares in Camaçari, Bahia. And here at the center you have Psidium larotianum with this new species of Nototriosa. When we look at that, what comes to my mind is the map of the biomes, the vegetation in Brazil. So when we changed that map for this one, you can see here these species are basically associated with Atlantic forest formation, while this one is probably more under the influence of Cerrado, even though we have Atlantic forest incursions here. And the thing now is, for me, is what is the natural history of the genus Nototriosa? For me, those are hypotheses. I think they might have been historically associated to the Atlantic forest by the distribution we've seen so far. And also another hypothesis is that the species from Cerrado are more recent. As we know, our neotropical forests tend to be very ancient forests, while our savannas are younger formations. So the species from the Cerrado should be more recent. Another thing that should be addressed is the gall morphology. Are there selective pressures over gall morphology? So I don't think so, because they look very, very similar and might have a different evolutionary history than the galls, for instance, the Quercus galls in Europe and other well-known examples. And I would like to end by thanking you so much for being here and paying attention, for the financial support by FAPERJ, the organizing committee, the amazing collaborators, and all the institutions and groups of researchers that support our efforts. Thank you very much.
Hello. Thank you for the presentation. And now let's move on to Barry's presentation. I forgot, but feel free to make questions on the chat. And in the end of the 3 presentations, I'm going to read the question and comments from the audience from them to answer. Mary?
Yes. Okay.
I will share my screen.
Ah, okay. Yep. So are you sharing it from your end?
Yes.
Oh. Ah.
Can you see?
Yes, I can.
Okay. You can begin. Thank you.
So, oh, great. Yes, I'm going to give a talk on the interaction between galling wasp, Leptocybin inversa, and its eucalyptus hosts, and particularly the Eucalyptus camadulensis, the river red gum. Eucalypts are a very important species in Africa, and we have 50% of the plantations, forest plantations in Greater East African region, and 25 in Kenya are eucalypts. And eucalypts are commonly planted also in private woodlots, so everybody doesn't want to be left behind. And eucalyptus forests in Kenya are dominated by Eucalyptus camadulensis, grandis, and saligna. They are also major source of fuel for curing tea and tobacco, as well as a source of construction poles and posts. But the major problem that now we're having as we go into plantation forestry are pests, especially the invasives that are now becoming common. And one of the common pests, the common pest is the Leptocybin inversa, which actually has invaded almost the whole world, and it moved quite quickly in the whole world. And by the time I was starting this work in Australia, the Australians didn't know much of the insect, and as much as we were making claims that it was of Australian origin, but the work we also did that is associated with this, related to this, showed that chances are very high that is from Australia. And the literature points to the northern part, which is Queensland, but our publications indicate that it could have actually come from the southern part of Australia. Our models showed that. And this makes the eucalypts look very unhappy from the photos that we see. Those are galls on the right. And those are actually several galls coalesced together into one huge lump. And the plant on the left has leaves quite reduced because of the number of galls that are on that plant. So I'll go next. And this is what the canopy looks like. It's actually a closed canopy, different from the canopies that we see on normal eucalyptus canopies. And the talk that I want us to have today is about the behavior of this insect. And the question that I posed to us today is whether the invasive Leptocybin inversa its behavior of indiscriminately laying eggs would lead to its end. And this is based on the sorry, back. Oh, this, if we were to base this on the female preference level performance hypothesis that proposes that a female should identify host plant modules and locations that would best support larval survival before depositing their eggs to maintain optical fitness of the species. And this should be seriously so for the galling insects, because the larvae inside the galls are immobile. And so there's supposed to be a lot of precision by the mothers in choosing a host that will really improve the fitness of the organism. So next. And what we did is we got seeds of Camerulensis from Australian Tree Seed Center in Canberra, and we selected 29 genotypes that represented the identified 7 subspecies. Originally, this was thought to be just one species, but the research has shown that there are actually 7 distinct subspecies. And so we got representation of subspecies acuta, arida, Camerulensis, minima, obtusa, refulgens, and simulata. That's classification according to MacDonald, 2009. And the genotypes were exposed to the wasps in nursery, common garden arboreta in Kenya, Africa. And also we observed and counted the oviposition that were being done by adult females under microscope in the laboratory. After that, the plants were also allowed the plants in the arboreta and in the nurseries were allowed to develop galls so that we could compare the oviposition and the galling. And we also did analysis of the terpenoid profiles of the leaves, and this showed that there were actually different chemotypes. As we can see, the 7 subspecies are separating well, and so our expectation probably would have been that the terpenoids would be important in guiding the adults into choosing a host. Next. And so this is what we were able to see under a microscope, the adult female putting the eggs into the soft tissues of the plant. And then we were able to see ovipuncture marks and actually counted them, the ones that were put under microscope and the ones that the tissues that we collected from the field, we would put them under microscope and count the ovipunctures. And next. And when we looked at the oviposition in different trial, different biases, we had no choice in the laboratory where we got soft tissues of the plant, so many of them put together in the petri dish, and then individual shoots in the petri dishes suspended in agar, and we then counted the number of ovipunctures that we were able to see. We also harvested soft tissues from the nursery and observed them under microscope and counted the ovipunctures. Also, in the garden arboreta, we harvested the soft tissues. And at this point in the beginning of this study, we still had very high infestations, and we just the arboreta were just exposed to natural infestations. It wouldn't take long before they came in, and that was the case with the farmers in Kenya. Their nurseries were heavily infested, and so it was very easy for us to just have the plants growing out, and the insects would come on their own and infest. And when we looked, so back at the back. Yes, and when we compared the levels of oviposition, the only ones that appeared to be having fewer oviposition in the upper chart was the ones with no choice in the lab, but then the rest don't seem to be different. They're significantly not different. And these are 29 genotypes, but collapsed into the 7 subspecies. So they are replicates. Some have 5 replicates and others have 4 replicates. So next. And we also looked at the intensity and incidence of the position in the lab, the one that we were able to watch and count. And they also don't look different, significantly different. And so the insect appears to lay eggs on the different genotypes equally. Next. And then when we went back to the field and in the nursery, the part of the plants that we had left to develop galls, then there appeared to be a lot of differences in galling. There were some subspecies that had very few galls, both in the arboreta and the nursery, especially the subspecies simulata and refulgens. But we had some subspecies that were highly galled and were looking very sick, the Camadulensis and minima, the subspecies. Next. And we tried to correlate oviposition and larval development. And when we talked about the intensity, the numbers, in the chart to the left or to my left, then there doesn't seem to be any correlation. But when we looked at the incidence, then there appeared to be a negative correlation, which means that the ones they preferred to lay more eggs on are the ones that would develop the least galls. Next. And then this appears that the disparity in the host acceptance for oviposition and larval survival on Eucalyptus camadulensis could spell doom to the survival of the invasions outside the native range. And this loss from lack of discernment for resistant hosts could lead to low populations of the insects. And this loss could further be aggravated by several natural enemies that have since been found associated with the pest in invaded range. In Kenya, we have identified 2 natural enemies, parasitoids that are found in the galls. Instead of the adult pest wasp emerging, we have the parasitoids emerging. And the galls have become rare in areas with known previous heavy infestation. And this has partly challenged our research work because we are unable to find the wasp as we used to do. Previously, we just go out to the field, collect twigs, and when we brought them to the lab, we would get enough wasps to do bioassays. But right now, we can't get enough wasps. We find a gall here and there, and so they're becoming quite rare. And then we are believing that the behavior of the adults is being detrimental to the populations of the insect and might really interfere or might really reduce the infestation levels. And probably then we don't need to worry so much about it as a pest because probably it will finish itself by not discerning what to lay the eggs on. Next, okay, and thank you so much for your audience. And in Swahili, we say asante. Yeah, great.
Thank you, Mary. Great presentation. So now, Christian's presentation.
Use the share button. If I have a problem, I will ask for assistance because okay. I can send also the PowerPoint to you. So let me see. I will press share, and then I have to press the screen or the window that I want to share, right?
Right.
Okay. Share screen. Share screen, and now I will select this. Okay, here we go. Are you able to see the screen now?
Can you see?
See? Can you see a picture of a sea that I am projecting now in the PowerPoint?
Yes.
Okay, so you're seeing the presentation. Okay, awesome. Okay, thank you so much to all of you that are organizing this meeting, to the Neotropical Group for the study of the galls. And what I'm going to show you is a little work that we were doing before the pandemic and was interrupted by the pandemic, but it's about the chemical changes that are found in host due to the Cecidology process. And my co-authors are people from the chemistry area. Yes?
Christian, can you put the presentation on full screen?
Full screen, okay. Here.
No, it's not full screen.
Wow, that's strange. Let me see. I was in my computer. I was seeing it in full screen, but I don't know why it didn't. Okay, I will try again. Share screen, select screen, full screen. Okay, there you go. Now you will see my whole computer, and if I project this, you should be seeing only the slide, right?
Okay. Yes. Yes.
Fine now?
Yes.
Okay.
Yes.
Okay. So I will start with this picture, the cartoon of these kids with the tree and the house in the tree, and the idea of that insects or other organisms, not only insects, bacteria and also nematodes, that are able to have their houses in the plants is a great phenomenon in biology. It's amazing. And when I was studying and I realized this kind of way of life, my mind exploded, because I found it really, really cool and interesting to study. And one of the questions that we have, and these are a photo of different galls from plants that we have here in central Chile. And one of the questions that you will come across to you when you are interested in this kind of insects and the plants that they live is that they have so many different shapes and so many different of phenotypes that they induce or trigger in the plant. And this is one of the dimensions that is really a matter of discussion until today. And so many beautiful works that the colleagues have been shown in this lecture are showing these different venues of changes, chemistry, morphology. And the changes in plant phenotype during the process of cecidogenesis can be divided in two broad categories: the morphology and developmental changes, and the differential expression or changes in the chemistry or biochemistry of the plants. And this can be related to the compounds that are kept in the plant tissues, and also in the compounds that are released to the environment, the volatinum or the volatile organic compounds that the plants release to the environment. And this last kind of changes has been less studied in general. And the hypothesis related to the changes that plants experience during the attack of a parasitic organism like a galler have two main also groups. One are the people that say that these changes are hacked or driven by the animal, or they're associated. They have been found, for example, different symbiotic microorganisms that are also responsible for some of the changes that you see in plants when they are attacked by gallers. And also is the hypothesis related to that this is a result of plant defense reaction. And most likely it's a mixture of both, because you can see different expression of the changes that construct the gall and expression of volatiles that most likely can be the result of both changes. Recently, 3 years ago, Borges made this awesome, very, very detailed review on what's going on about the volatile compounds when a plant is attacked. And she made this graphical abstract of the problem, and she said basically that it can the the host can be releasing volatiles to attract the enemy of the galley. For example, nematodes or parasitic wasps. By other ways, also the parasitic insect or their associated can be suppressing certain kind of volatiles that can be an alarm towards these predators, and the plants will release less volatiles. And most often, based on the papers that were available 3 years ago in this review, they found that basically in most cases, the plant that when get attacked, is reducing the volatiles in the number of cases that was studied. Also, there are cases where the galler is able to modify the volatiles and also to use them to protect themselves towards their natural enemies. This will include not only parasitic wasps or predators, but also some herbivores like mammals. There are some cases where aphids are able to suppress volatiles from certain plants, and in this way they avoid to be eaten indirectly by squirrels or other mammals that go to feed on these galls. So there can be different alternatives to these processes. And the problem is basically that there's little amount of evidence to draw a pattern or a general hypothesis of what's going on here. What is known is what I'm showing here in this graphical abstract, and also it's known that these volatiles can derive from different structures or organs of the plants that for sure you all know very well. I will now continue illustrating this problem with the case of the Aplopappus foliosus truth in central Chile. This is the map of the biodiversity world hotspots, and this is changing from year to year, but more or less there are several hotspots in South America, and one is in central Chile in the area that is called the Matorral Biome. And here is a map of South America where I'm pointing the area where we perform our studies. All this story started while I was doing a project related to the insect associated to the Aplopappus genus, this trope that I mentioned. And these plants are very interesting because there are more or less 37 species in Chile, but they are very good for generating hybrids in the altitudinal gradients and also along the longitudinal distribution. So you can see herbivorous communities changing from host to host through these herbivorous phenotypes. And when I was doing this study, I realized that there was these swellings in the bushes. If you see here, this is the Aplopappus foliosus, Asteraceous trough, and in the Mediterranean xeric matorral in central Chile, in the coast, you see this kind of cliffs, little cliffs that fall in the sea, like the one in the first photo that I was showing. And it's a very dry area where the soil is very with a lot of clay. So it's difficult to generate a good soil for the plants to grow. So the plants live in a very stressful condition, and it's often to find galls in different plants, either even herbs are galled. And this is one of the galls that I'm showing you here. And when I faced to this problem of the galls in the Aplopappus, I hit my head with a wall because the animal that was inside was unknown. I contacted Professor Gagné and other specialists, and they recommend me to contact Professor Gabriela Sidmaya, and that was a very fortunate thing to do because she was very kind and generous and helped us to know what is this problem, what's this insect that was making these galls, and thanks to her help, we were able to identify this as a gall midge. It was a gall midge that was not known yet. Here you can see the gall growing in the apical branch of this shrub, and this is a male here in the side, and in the other corner you can see how this is the structure of the gall inside. The larvae are sharing a chamber. This is very interesting. They share this chamber, and inside there is this fiber that is also trigger or a product of the gall formation. So this was a new species, and we worked together with Valeria to be able to identify this. It was very exciting work to do, and we were able to describe this as a new species of gallmich. And also, what is interesting is that this gallmich is not alone. Is always attacked very heavily by parasitic wasps. We reached to a level of knowing that this is a platygastrate species, but we need to contact people that work with these groups to know to be able to identify it. And this is a heavy attack. Most of the galls, you see the galls like this with the larvae will be, I mean, most of the half or more of these larvae will be parasites. Will have will be parasitized by this platygastrate. And so now we reach to this second chapter of the story. We were interested in to know, as parasitic wasps are very good for following volatiles as their key to find their host, we were interested in to know if the process of gall induction or gall formation may trigger changes in the phenotype, in the volatile profile of these plants. So we have these branches here in the upper part of the corner. You see the healthy apical branch, and this purple circle means turkey oven bag. That is a plastic that you can put it in the oven. So used in North America, it's used for cooking turkey or something like that. And you can use it for scientific purposes. If you want to have a bag that have no traces of chemistry, you burn it in an oven a little bit, and then you can use these bags and put it over the branches, and with a headspace system that is a polymer connected to a pump, you can start pumping out the air, and the bag have another role on the other side, so you can collect the living set of secondary chemistry that the plant is releasing in that particular moment. So you sample this, and then this column, that is a glass column with a polymer inside, you can dilute it with some solvents, and then refrigerate the sample. And after that, you make some chemical analysis with GC, HPLC, different machines, and you can have the profile of volatiles either derived from branches that have no attack, no evidence of attack, either little buds or galls that are really formed, or versus the situation where you have this gall niche developing inside this structure. And we did so. We collected in the field in living plants without coding or anything. These branches are connected to the plant, and then we took this to the lab, and then we have these compounds, and there are some serious number of compounds here, and these are aromatic compounds, monoterpene hydrocarbons, fatty acid derivatives, and oxygenated monoterpene and sesquiterpene. And what is interesting to see in this list, you will see in the next slide, is that the proportion of the main compounds that we find in the mixture changes when the plant is under the gall situation. Here you see in purple the healthy apex, and in this kind of pumpkin yellow color is the gall. And here are some of the compounds that were the most relevant based in a principal component analysis that we did. And this is the proportion what we found, and is very clear here that GALs have different proportions of some compounds. For example, cement is more present in the healthy apex, and for example, limonene is more present in GALs. In the case of cement, is called as a compound that helps sometimes plants to be able to defend against chewing herbivores. Meanwhile, limonene is a compound that is quite important and is the main attractor of parasitoids. And not only that, is that we found evidence from other systems where the platygastrid parasitoids, that are the parasitic wasps of these gall midges, platygastrid parasitoids that were parasitizing other gall midges are attracted to limonene. So this is exciting in the sense that it may have a sense as a semi-chemical signal. We were conducting the early steps of white tube olfactometer collection and observation of the behavior of this wasp when the COVID problem kicked us out of the university. So we will have to continue that when we will be able. And what is interesting then is that the set of compounds that the plants have to release as volatiles are often not only addressed to a main goal like defending against the gall midges or attracting parasitoids, but are a set of traits that are exaptation or derived from other initial functions, as for example UV light protection and the resistance of the plants against evaporation. So these patterns are really interesting in xeric environments or dry environments are interesting in this sense. And if the gall insect is able to hack the plant or to make it produce compounds that are good for them, that will be a way to explain it. In this case, we think that the case will support at least or data support that the plant is trying to reduce the damage on the reproduction that this cecidogenic insect is inflicting to this host, because as you see these shrubs, when there is a gall on the apical part of the branch, there's no flowers, so that branch is lost for reproduction. In that way, the gall midget is reducing the reproduction success of the plant. And this is a venue of process that we're exploring also with other species of Aplopapus. As I mentioned earlier, there is several species of Aplopapus just in the central region where we live. You can travel to the mountains and find other species that also have these galls with a very, very similar to Aplopapus, or other species of Aplopapus that are from the mountains also have these galls. So it's very likely that either this gall midge is using several hosts, or there is more species happening, and we're very interested in to continue unraveling this problem, and also to explore the volatile changes if there is anything in this case. And again, I would like to thank Valeria, and I tell all of you that she was very, very generous, and I consider her my professor. And we hope that we want to continue working to know what's the rest of the gall midget that we have in Chile, because there's much to do. So thank you all for your attention. Obrigado. Thank you. Muchas gracias.
Thank you very much for your presentations, Christian, Verin, and René. Now we are going to read the questions of the audience for you. Okay. Wait a second. Okay, so the first question is for René. René, do you know What determined the gall color, red or green in the system?
Yeah, no. We've tried to, relate it to parasitism, as is usually found in literature, but it did not correlate. These galls are attacked by Galeopsomia wasps, and as far as I know, it's the only one that attacked that gall, but I'm sure there are other organisms, but I didn't account them. So maybe, other organisms may affect. I know there's an inquilin because I've seen structural changes sometimes, but no, we don't. It may be, light or stress or other sources of stimulation.
Okay. So the next question is for Berin. Berin, can you turn on your camera, please? I don't think she's. Wow. So I guess let's move on to the next.
Let's see if she, not sure if she's listening. So maybe you should ask Christian a question, then we try to reach her.
Okay. So Kevin Float asked to Christian, is the suppression done by immatures?
Yes, absolutely. Because when we, at least the evidence we have till now, we followed the development of these galls first 1 year to know the exact phenology of the species. So we know quite accurate when the gall was well developing with the larvae inside. So most likely these are the ones that are conducting the main changes, at least in volatiles, if there are some influence from these guys. What is interesting also is that we have additional evidence regarding the microbiology of this system, where we have found fungi associated with these gall niches, and these fungi are releasing some compounds that may be related also to these changes. But we have to conduct HPLC to these colonies that we started developing. But we can access the lab yet, so we will have to wait.
So we have a question for you, Berin, from Rosi Isaias. Berin, are there any native species to substitute plantations of eucalyptus in your country?
Not sure if she's listening.
Berin, are you listening?
I think she sent a message in private chat.
Mute. Oh, great.
Yes.
All the eucalyptus in Kenya are not indigenous, but we have different species in different regions. Yeah, and related to that, or is a question that I've seen in the comments, somebody asking about globulus. So globulus is also very rare in Kenya and only planted in the highlands, and all these are restricted to plantations, not in the wild. We don't have any wild eucalypts in Kenya.
Okay. So the next question is also from Rosie. She asked to Renee, do you think these similar galls involving congenerity, speciation congeneric species are good models to evaluate host?
I think I've seen the questions. So there's a word missing, but it's okay. I get it. Maybe, Rosie, if it's actually the case of co-evolution. If the insects have co-evolved with the host, yes, they try to study the phylogeny of both insects and host plants. Should complement each other and sort of work together on a time basis and on a speciation basis. But if it's not the case, no, it won't be informative for evaluating host evolution. We should, try to make a dated phylogeny of both plants and the insects to know if the events of speciation occurred by the same time. Who knows?
Valeria Sidmaya asks for Christian. Are volatiles more efficient in arid areas?
Yes. Regarding the ability of little compounds to disperse in the air, it's known that when the weather or the environment is rather hot than cold, it's easier for these compounds to travel farther. So you can imagine a situation where the plants that release volatiles in dry or not so cold environment or so wet will be more efficient or more able to use these signals as attractant to predators or parasitic wasps. It's something that you can hypothesize. Absolutely.
Jessica Howard asks to Dr. Otieno, how do you think lepidoptera invasion has spread so quickly throughout the world? Does it move through the lumber trade or other agriculture products?
Yeah, we only can suspect because we don't have any proof of the movement. So locally, especially within Africa, we see a lot of interactions in the neighboring countries, and people move seedlings from here and there, but the chances are not there of it moving in the lumber because this is an insect that attacks the growing tips of the growing plants. And so if you brought in the lumber that is dead, you probably won't have much because it grows on the foliage. But internationally, we see a lot of movement, and the adults of these insects can stay for more than 7 days in some cases. We are imagining that probably people can put it in a packet in their suitcases, especially from Australia. And because the trend has been, the invasion that we got here in Africa began from South Africa, and most of the travel routes to Africa from Australia is through South Africa. And this has happened with most other invasions that we have. Majority of them are first detected from South Africa. So probably human beings not knowing that they are bringing in the pest could also be one of them.
Okay, so the next question is for René. So the question is in Portuguese, but I'm going to translate for the audience. The distribution of the plants is also strict. Your microphone is off, I guess.
I'm sorry. I was afraid that it was capturing the sound around. Sorry. It's not. These plants occur throughout Brazil. I mean, Psidium myrtoides from northeast to the south region. Catleyanum, the same thing, and Laroteano is more. It's also widespread, but also on the center-western part of Brazil, but not all the way down to Rio Grande do Sul. So they are widespread, not restricted. There might be a slight problem because last year, by the end of the year, the specialist on Psidium species told me that the plant I thought it was Psidium myrtoides is actually Psidium corinanthum, which is restricted to Minas Gerais and Rio, but not sure yet. So if it's the case, only Psidium myrtoides, or I mean, Nototriosa myrtoides would be restricted to a species which doesn't have a wide distribution in Brazil.
Okay. So the next question is from Bruno Garcia Ferreira. Christian, do you believe that volatile production was primarily a defense against the galling activity, or is an induced feature which is adapted to the inducers? Your microphone is off, Christian. Sorry.
Yes, that's something that we have to test experimentally. But if you make a good guess or hypothesis regarding that, at least if the plant, when it's induced the gall, change the proportion towards the compounds that are more attracted to the parasitic wasp, I will suggest that this is important for the plants than positively for the plant than for the gallers. The gallers, as I mentioned before, get heavily attacked by parasitic wasps. So it's likely that this is a way the plants are attracting by induced defense, indirect induced defense mechanisms. That will be my hypothesis in this case.
Okay. The next question is from Jose Isaias. Betting, are there hybrids or species more susceptible to leptocyte invasion?
Yes, Jose. Even within the complaints that I was studying, there are some subspecies that are highly susceptible. The galls form. I only compare between oviposition and gall development, but some are more preferred. There are others that don't develop galls at all, while others are hugely galled, and even growth becomes an issue. Apart from the Eucalyptus camaldulensis, we have grandis, which is also very highly susceptible. Some provenances of grandis.
I think she might have connection problems.
Yes.
Sorry?
Your connection was...
Yeah, we didn't. We weren't able to listen to the last part.
Did I get lost?
Yeah, a little bit, but only at the end.
Oh, so should I have a go again?
Yeah.
So
okay.
My answer was that there's a lot of variability even within the same species. Actually, even just different provenances could be galled differently. Some have high galling and others wouldn't. And even in the case of Eucalyptus camaldulensis, the different subspecies were quite variably galled. Others were not galled at all, while others were heavily galled. So, yeah. Did you hear that?
Yes.
Hello?
Yes, we did. I think she lost the connection again.
So the question for Christian is from René. How much volatiles of these galls produce? I remember Adriano Cavalleri's talks yesterday, and he's saying they could actually smell trips galls. How variable are volatiles across different taxa?
Yes, that's something that you can experience too in this system, because when the plant get this apical structure of the gall, the fragrance is subtly different. It's more rancid. It's like a rancid woody smell. While the plant, when it's healthy, the branches smell like lemon and pine. Is totally different smell. That's a good observation. The colleagues said you can perceive at least in this system. In other system, I don't know, because for example, there are some galls for example, in Boldo that are made in the branches, and you cannot tell by the smell because the plant is so pungent, the smell, that you cannot by your senses see the difference. I don't have the data right here of the parts per million or the amount in quantities to tell you right now exactly, but at least my perception in the Ablopapus system is that the gas smell quite different, in fact.
Nice.
So the next question is for René from Denis Oliveira. In addition to Rosie's question, do you think these galling insects can switch to other species into host genus?
Well, it's hard to. It's hard to affirm that, but I believe they can shift, or if it's fruit of coevolution, most likely they can't. But if not, they would be. I have to mention that Ivonetti, when she was working with those galls here in Goiás, there are actually two varieties of Psidium laroteanum. There's this glabrous one, and the other one is, I don't know, it has indumentum, dense, sort of dense indumentum. They are different plants on the field, and she had the hardest time because she's very meticulous, and she went crazy because she couldn't separate the galls. And then Daniel said probably they were the same species, but he's not sure yet. So I don't know, maybe we are witnessing an speciation event, but who knows? We have to, do the phylogenies of the insects to take a guess.
Okay. So the next question is from Christian Alfonso Milagra to Berin. Excellent work, Berin. Have you tried to evaluate the host of orange's influence on the oviposition preference of adults?
No, not really. Not really. In the current studies, what we did is we got the adults out of their hosts for after some time, and kept them in cold and starved them for a while, so that we don't. I don't think we've done that. No. But we tried to take care of that in this study because we were suspecting probably that would influence them. So that's why we kept them away from a host for a while and kept them in the cold. Probably the next thing to do, I guess. Yeah. Okay. Thanks.
So the next question is, okay, wait a minute. So for Christian from Lubia Maria Guedes Garcia. Christian, have you studied the influence of the parasitoid on the volatiles profile? If yes, have you found any difference between gall, non-gall tissue, and parasitized gall?
Yes, that's an excellent question. In fact, for example, in our system, we have not the chance to evaluate this experimentally, but there is known in parasitoids that attack aphids. It has been found that the parasitic wasp can transfer virus to the aphid that can, through the aphid, pass to the plant and change some features of the volatile. It's really incredible. And we are hopeful to be back in the lab, probably the second semester of 2021, to test how these processes can be going on in this system, because at least our impression is that mainly the process of the growing of the larvae inside the gall is related to these changes, but you have to evaluate that too. It's something that is interesting to test, but it cannot be done through the process that we did in the field, but having an experimental setup in the lab or a more controlled situation where you can be sure that the gas were not parasites, parasitites. So that's the problem, that we have to do that kind of experiment now that we cannot access to the labs. We can go to the field, but we cannot go to the lab. So absolutely.
Okay. So the next question is from Bruno Garcia Ferreira for Berin. Are there studies in biological control of leptospira invasion by parasitoids?
Yeah, there are studies, and most of the studies are being carried out in Asia. We haven't done any in Kenya, but we have a lot of studies also in South Africa. But in Africa, I think the earliest that was done in Israel, and to identify the biological control, and the parasitoids were still, from Australia, but we attempted to introduce them even into Kenya, but we were not successful. Why they didn't succeed? But right now we are seeing something that we don't have any history of introduction, just appearing, popping up, and now reducing the populations.
Okay. We have a question for René from Iguaba, Arriola. René, why do you think that the Atlantic rainforest is the center of dispersion of Novelties? Cerrado is another biome and is a natural distribution area of the species and others. Precision.
Yeah, Igor. Actually, the most recent data showed that the assembly of plants and animals that occupy the neotropical savannas nowadays are actually younger than the assembly of the forests, for instance, like the Amazon or the Atlantic forest. Actually, the Cerrado has an older geology history, but fossil records show that it's a younger biome. I mean, the way we know it, it's like 23 million years old. Like, and the rainforests date back to 60 million years back. And I think that might be the case. I mean, it's a hypothesis, but I think, if I'm not mistaken, the center of diversity of Myrtaceae in Brazil is the Atlantic forest. So as they associate with Psidium, which are Myrtaceae species, it's a hypothesis that I came up with. So we have to test it.
So Denis Oliveira asks to Christian, could the high local plant diversity works as a natural resistance for galling attack once the huge range of volatile emissions could not be effective to attract specific galling?
Yeah, that's a good idea. We have no idea of experiments that have evaluated that, but there is evidence of diversity of compounds, of chemical compounds that are linked, like correlated positively with the amount of diversity of plants. So that may give support to Dennis' idea that when there are so many species, plants can have to work harder to differentiate in the fragrances, and that may be the case for indirect defenses. For example, of the compounds, the mixture of compounds are released, because in most cases you don't have new compounds appearing in the plants, but the composition get changed. So I will suggest that, based on the evidence that show that when you have more diverse a community that's more diverse, those plants have more diversified compounds, that this idea have total support. But it had to be tested in the case of the plant gather other trophic levels system to see if this is the case too. Yes.
Okay.
João?
Yes.
Maybe I think we should, unfortunately, end the session because we are already a little bit late. And then I'm gonna apologize for you guys for doing this, but we can go back to the chat later and have this pleasant conversation. Sorry.
Okay, no problem. Thank you.
Thank you.
So now we're gonna finish, and
Thank you.
thank you. Sabrina is going to.
Thank you all for the amazing presentations. So now we will have a 10 minutes break, and we will come back with the third session, which is entitled Gauss from Ecological to Molecular Complexities. So see you soon.
Hello, we are back to the third session of the symposium. Welcome again. My name is João Victor Nunes. I am a biological science undergraduate student at the State University of Feira de Santana, Bahia, Brazil.
My name is Sabrina Vasconcelos Carão. I'm a biological science undergraduate student at the Federal University of Espírito Santo. We are members of the chapter Iniciação Botânica, a group of young botanists affiliated to the Brazilian Botanical Society. 4 Brazilian universities from 3 Brazilian states are involved in this project, whose aim is to introduce biology students in botany subjects, promote and support scientific events. This is the first event that the chapter is hosting this year, and I would like to invite all of you to follow us on social media, Instagram at Iniciação Botânica, because we have exciting news and events upcoming.
So we are going to be the mediators of the sessions about goals from ecological to molecular complexities. So we will have 3 presentations with 3 different speakers, and I'm going to introduce each one of them. So the first speaker is Dr. Kevin Float. He holds a BA in agriculture from the University of Saskatchewan, 1983, master's degree in biology, University of Saskatchewan, 1987, PhD in zoology, Northwest Arizona University, 1992. He is an expert in crop and livestock entomology, insect microbe, symbiont interactions, biological control of insect pests, non-target effects of chemical residues in livestock dung. And the second one is Milton de Souza Mendonça Jr. He has a permanent position in the Department of Ecology at the Federal University of Rio Grande do Sul, Brazil. His research involves ecology, entomology, and also astrobiology in his spare time. Current projects include the development of arthropod and plant interaction networks, the study of ecology and the restoration of pastures and forests, and the link between ecology and astrobiology. He has recently had a 1-year sabbatical at UCLA from 2019 to 2020, and a short stay at the University of South Florida in 2012. The third speaker is Jack Schultz. He is a distinguished professor of entomology emeritus at Penn State University and currently research professor at the University of Toledo. So feel free to make questions during the presentations on the chat, and in the end of the 3 presentations, Sabrina is going to read the questions and comments from the audience for you to answer.
So can you see my presentation?
Yes, Kevin.
Okay, thank you. First of all, thank you to the organizers for inviting me to present here today. I'm up in Canada, where this little red dot is, and I understand a number of people are down here in Rio de Janeiro. So if you look really hard, you can see me waving at you from across the globe. And today I'm going to talk about a project that my wife, Rosemary de Klerk, and I have been working on out of the Lethbridge Research and Development Center in Lethbridge, Alberta. And it's about the interaction between symbiotic bacteria, a wasp, and possible speciation. So the name of the wasp is Elicidia pilicillae. It's a European species in family Cynipidae. It forms blister-shaped galls on the leaves and stems of hawkweeds. And these are species of plants in the genus Pelicilla. Now, some of these hawkweed species have invaded North America and are now problematic weeds. So the interest is in using this gall-forming wasp as a possible biological control agent to try and suppress populations of these invasive weeds in North America. And on the photograph, you can see an ovipositing female. In the middle photograph, you can see the blister-shaped galls on the midrib of a leaf. And then finally, on the far right, you can see the dandelion-shaped flowers of the hawkweeds. Now, back in 2012, a master's student by the name of Chandra Moffat traveled to Europe, and she collected field specimens of this wasp from different geographic locations. And she brought these specimens back to Canada, where we did CO1 barcoding to look at their genetic diversity. And we confirmed that everything she collected was the species Alicidia pilicillae, based on the relatively small amount of genetic differentiation between specimens. So in green and in blue, these are all specimens of Alicidia pilicillae. And then for comparison here in brown, these are different species of Alicidia. And obviously genetically, they are very different from the other material that Chandra brought back from Europe. But one thing we did pick up on is that there's two genetic clades within the species. So here we have a branch point with very strong support showing one clade that I'm going to call biotype 1, shown here in green, and a second clade, what I'm going to call here biotype 2. Biotype 1 was reared in the field from different species of hawkweed, excluding Pelicilla officinarum. Biotype 2 was reared from the field only from Pelicilla officinarum. So there's genetic differences between these biotypes as well as host plant usage differences in the field. And two specimens, we don't know what the host plant was, so I haven't indicated that. A third specimen we think was reared from a hybrid between Pelicilla officinarum and a second species of hawkweed, C. spitosa. Now, Rose established 6 populations of these different biotypes in her lab in Lethbridge. So she had 4 populations that genetically were biotype 1 and 2 that were genetically biotype 2. So the biotype 1 populations were from Zweibeck, Strasny, Cisnoka, and Milkow. And the biotype 2 populations originated from Southern Europe as well as Milkow. So to be clear, there are 2 biotypes from the same location in Milkow, but they have different host plant usage. So these biotypes can co-occur in the field on different host plant species. And then over the course of a number of years, Rose was able to show very clearly that the biotypes differ in life history traits. So biotype 1 only produces 1 generation a year and then has to enter a mandatory cold period. Biotype 2, you can rear continuously in the lab without a cold period. When the females emerge, they have their full lifetime complement of eggs in their abdomens. So you can dissect the newly emerged female and get a measure of its potential lifetime fecundity. And when Rose did this in her lab, the biotype 1 females, for each of the 4 populations that she had roughly 100 eggs when they emerged from pupae. The number in brackets here are the number of females that were dissected for each population to get the average value. And in the biotype 2 populations, those females had roughly 70 eggs upon emerging from pupae. And then finally, the biotype 1 populations had both male and female individuals, whereas the biotype 2 populations only had female individuals. And again, over the course of a number of years, Rose monitored the sex ratios of these lab populations. So we have 3 populations of biotype 1 in this chart. In each of those 3 populations, in each year that she collected data, there were always males and females. And the number in black font in square brackets are the number of individuals upon which Rose determined the sex ratio. And then we have 2 populations of biotype 2. We never had any males in those lab colonies. But we have a very interesting case with one of the biotype 1 populations this one from Zwiebek. Initially, there were males and females. And then in 2017, when Rose collected data, of 105 individuals that emerged into the lab cage, only one was a male. So this Zwiebek population seems to be undergoing a transition to become parthenogenetic. Well, this is where I come into the story. In my lab, we've done a fair bit of work on symbiotic bacteria associated with insects. And one of the bacteria which gets most of the fame is Wolbachia. And if you haven't heard about these symbiotic bacteria, they live inside the cells of the insect host, and they really cannot survive outside the cell for any period of time. So these are obligate intracellular symbionts. They're very common. They're one of the most successful parasitic life forms on the planet, perhaps. They are known to occur in anywhere from 20 to 70% of all insect species, as well as other arthropods, such as isopods and spiders, as well as in certain types of nematodes. So they were a very successful type of bacteria. But because they cannot survive outside the cell of the host, transmission is typically vertical, from an infected female to her progeny in egg cytoplasm. So here's a photograph of a wasp egg, which has been stained to show the DNA of the Wolbachia. So each of these little tiny white specks corresponds to a single Wolbachia bacterium in the egg cytoplasm. I read so much about Wolbachia, I don't know what to believe, to be honest. But my impression is that Wolbachia is like a superhero with many powers. It can fundamentally alter the biology and life history traits of the host. It can also alter the reproductive phenotype. I could spend hours talking about it, but for the purposes of our talk, what is important is the ability of Wolbachia to induce parthenogenesis in haploid diploid species, like the wasp that we're studying. So how does this occur? So let me just begin with a review of haplodiploid life cycles. So if a wasp lays an unfertilized egg, it remains haploid, it becomes male. If the female lays a fertilized, or if the egg is fertilized, it becomes diploid and produces a diploid female. But what is important is that once you have the egg, whether it's haploid or diploid, it undergoes a cellular division. And at that point, it produces a duplicate set of chromosomes. And then in the two new cells, each of them will receive one duplicate set of chromosomes. But if the female is infected with Wolbachia, you have that first cellular division, you get the duplication of chromosomes, but Wolbachia prevents the separation of the duplicate sets of chromosomes. So instead of each new cell getting one set of chromosomes, both sets of chromosomes stay in one cell. The other cell does not get any chromosomes and it dies. So what you're left then is with a diploid cell formed from an unfertilized egg, but because it's diploid, it becomes female. So that's how Wolbachia induces parthenogenesis. Well, because of our interest in Wolbachia and because other people had reported that it causes parthenogenesis in haploid diploid species, we went back and looked at the original material that Chandra Moffat had collected from Europe, and we tested it for the presence of Wolbachia DNA. So where you see the bold font, those were individuals that were tested. And if the font is in red, that's positive for Wolbachia. So in biotype 1, we tested a number of individuals for Wolbachia. Only one tested positive. And that was this unusual Zwiebek population. If we look at biotype 2, we tested a number of individuals. Only one did not test positive for Wolbachia. And this was the individual that we reared from a plant we thought was a possible hybrid of two Pilosella species. We also at that time had a colony of Alicidia subterminalis, which was known to be parthenogenic. And all the individuals of that species we tested were also positive for Wolbachia. So the message here is that there's a strong connection between the presence of Wolbachia and parthenogenesis in this system. So then we decided to take our story a bit further. We took 5 individuals from each of the 6 lab colonies that Rose had in her lab, and we sent them out for next generation sequencing. And then we used the results to determine the relative abundance of Wolbachia in each of the tested individuals. So for example, these first 3 bars correspond to biotype 1, so 5 individuals associated with each bar. The black dot indicates the relative abundance of Wolbachia compared to all the other bacteria sequences that we got back from next generation sequencing. So instead of 5 dots, we have 1 because they all overlap each other. So there's really no Wolbachia in any of these first 3 populations. The next 2 bars correspond to biotype 2, and the individuals tested all had very high levels of Wolbachia. And then we come back to this unusual Zweibeck population. 4 individuals tested very strongly for Wolbachia. The 5th individual had no Wolbachia. So here's what we think might be going on. Imagine an ancestral condition where Allocydia pellicella was not infected with Wolbachia. So you have bisexual populations, 1 generation a year, and they can form galls on many different host species. At some point in time, Wolbachia invaded some populations, leading to parthenogenesis. And with parthenogenesis, you have reproductive isolation. With the further passage of time and natural selection, these univoltine populations become multivoltine and they become specialists on Pellicilla officinarum. And we can explore that a bit more during the question session. Now imagine a second, more recent invasion by Wolbachia in uninfected populations, resulting in some populations remaining uninfected, but now some more recently infected populations becoming parthenogenic and becoming reproductively isolated. I think this scenario would nicely explain what we're finding in the field. So we have biotype 2 and biotype 1. They become reproductively separated quite long ago in evolutionary time. And that leads to genetic differences corresponding to the 2 biotypes that we can pick up with CO1 barcoding. But we also have this Zweibeck population, which is more recently reproductively separated from biotype 1. So genetically, it appears to be biotype 1, but it's becoming parthenogenic. So what happens with this Zweibeck? It could continue to evolve and become more like biotype 2, or it may take a different evolutionary path and become a biotype 3. We simply don't know. So I just want to finish up with a couple of take-home messages. Wolbachia truly is very common in many insect species, and it could have huge effects in our study systems, but many entomologists are unaware of the important role these symbionts may play in their system. So part of the reason I'm giving this talk is to simply increase awareness of Wolbachia and the role that it can play in insect biology and evolution. But also, if you happen to be studying an insect as a potential biological control agent, understanding how symbionts may be influencing the ecology and biology of these agents may enhance the potential success of your project. So I thank you for your attention, and I just want to acknowledge a number of people that were involved over the years and continue to be involved with this project. So thank you very much.
Thank you very much, Kevin. Really good presentation. Now we're gonna have Milton Mendonça presentation. Welcome, Milton.
Hi, how are you doing?
Yes, fine.
So do you hear me?
Yes.
Okay. Here we go. Let me share the screen for you.
Okay.
In just a second. Okay, so can you all see my presentation?
So we see the screen, but it's all black.
Oh, okay. Let me try something. Let me try sharing the. Let me share the whole screen. Is
Yeah,
that better?
now, yes.
Okay. So, hello everyone. Good afternoon, and thanks a lot for the invitation. Thanks to Jose and to everyone involved. Really happy to be here, sharing this time with you and telling you about some of the research we've been doing here down south. So I'm going to be talking to you about how to build networks out of goals, which usually go by the name of complex. See, it's supposedly a step further from tritraffic view we have of the systems, but really I'm just following on the steps of everyone that spent decades working on this kind of system. So I'm from the Federal University of the Rio Grande do Sul state in the south of Brazil, representing the ecology department. And I'm presenting also, which is something which is the result of the work of two of my colleagues, Carolina Silva and Fernando Luz. So I'm going to start by telling you that networks actually are a way to represent communities, biological communities. So we can, when you build a network, you can see richness, you can see composition, and you can see the interactions among the components of a community. So you can see the structure behind these interactions and the species. So it's not only knowing which interactions occur and which species interact, but from this you can also derive which determinants are important for this specific community, which factors affect how and why species interact. If you take a functional overview of this interaction as well, you can probably see there are forbidden interactions that will limit connectivity, which means some species cannot interact with others because of the way they're built, basically because of their biology. And by looking at network topology, I mean, by taking a step back and looking at the network as a whole, you can see structure, and you can probably think about stability and the coevolution of the whole community as well. So it's really helpful. And it uses what is known about networks in general from other fields of study as well, like sociology and even technological networks like transport and such. So there's lots of ideas around about how networks are built, not only from ecology, and we can use all that to our favor. So goals, as you know better than me, are really good for interaction networks. A treasure trove for network construction. You find the structure and you find lots of different species interacting there. One problem for the neotropics is feeble taxonomic knowledge on gullers and wasp parasitoids, of course, to speak of only of these two components. But with smart morphospeciation and genomic methods, we can go further with this. I think we should not be stopped by this linear impediment. And it has been done a lot for Europe and North America. We have lots of historic data on food webs, especially for sonipid gulls, for example, which we can see an example here on the right. Lots of different wasps attacking galling wasps. So we have a full food chain on a single gall sometimes. So this structure is identifiable and it is culturable. We can take it to the lab and it's made of plants, so it's kind of resistant. And you can find at least 3 trophic levels very easily. Not to mention all the other things that involved on a gall. Here you see on the left an illustration from a paper by me and Fernando Luz, in which if you think about all of the interactions between Cecidophages, which eats the gall, the inquilines, the kleptoparasites, other symbionts like fungi inside the galls, there's a whole world inside a gall. Here I'll be talking only about relationships between the plant inducer and the parasitoids. So it's really restricted right now, but I'm hoping to, and I leave this as a message for everyone. There's lots to do in relation to this other interactants as well involved in the gall. So networks with gall systems are a way of looking at these communities. And given the amount of species, the diversity of species we have around all around the world, there's really much to do about this. So I'm going to bring you here very shortly 3 examples with networks built from goals as starting points. I'm hoping to leave it clear to you how useful these goal systems are to understand networks. Networks can be tricky to build. They require, for some systems, lots of information. And for when you use goals, it's kind of easy to derive this interaction. So 3 examples, a little bit different in scope and aims. There are 3 different works we're developing. The first one starts from a single host plan in which we find many goalers, and also these goalers have each their own parasitoids and some parasitoids which are shared, which is kind of the main thing, the main question. And we looked for effects of goal structure on the network. So how would this functional coupling of goals and parasitoids relate to the structure we find in the network in the sense of who interacts with who? The second example involves 2 host plans and all the goalers present there and all the parasitoids we could find. The difference here is that there are oligophagous and monophagous goalers. So these 2 plants are very close, they're congeneric, they're very close phylogenetically, and some goal species, some goaler species are able to use both plants, and other species are able to use only one of them. So we wanted to know how this plant identity change could affect the structure of this network. And the final example is a system with multiple host plants. So all the plants we could find in a given environment with goals and their parasitoids. We were comparing 4 areas actually, which were under restoration, and comparing them to native vegetation. So the main question is about whether restoration would be working adequately for the system. And we were using goals and interactions to demonstrate that, and not only plant composition or vegetation structure. So our hypotheses are that the fewer the hosts, the more connected these networks would be, and the more complex they would appear to us, of course. So to summarize the methods, I'm also using red dots here, just like Kevin Flood did before me. I'm coming from the very south of Brazil, as you can see, and the northern part of our state has subtropical forests. We explore different forms of this forest, where our samples are really pretty much the same. It varies just a little bit according to the system, because sometimes we are looking for a specific plant, and sometimes we just taking every plant at any goal that appears. So it's usually time-based sampling. It's usually 60 minutes, 1 hour sampling on a given trail, not exceeding 200 meters, and that will mean a sample for us. So we search for specific host plant in the first case, for 2 conspecific congeneric host plants in the second example, and for any plant with goals in the third example. We just get the goals in the field, take them to the lab, put them into vials and bags, and wait for the emergence of any goalers or parasitoids. And we score them, we identify them to the best of our knowledge. And so we can build our network based on the identity of the plants, of the goalers, and of the parasitoids. So here you go. This is the single host plant network. I'm sorry, there's lots of information there. It's built on Guapira opposita, which is a Nyctogenaceae, very common here in Porto Alegre, in the capital of Rio Grande do Sul state. We sampled 1,762 galls in 6 Cecidology species. We found 741 parasitoids of 45 species. We find intermediate specialization levels, and that 21% of all the possible interactions, given this six galling species and 45 parasitoid species, only 21% of the connections were actually found, which means that only one in 5 possible interactions were actually seen. We tried different models. We tried to see whether the ecological processes predicting network structure would be neutral, just based on the abundance of each species, or special questions, which were whether they were present on a given set of trails or another. Temporal structure, which would mean whether parasitoids occur at the same time as the gallers or not, and morphological coupling factors, which prove themselves the real culprit. And we reported that in last year in a paper in ecological entomology. We found that gall thickness interacting with parasitoid ovipositor length was the main question. So there are forbidden interactions. Very small parasitoids cannot attack very large galls. You would probably say that it's pretty obvious, but we could demonstrate that that's exactly how it happens here, and that's the reason why the network is as such. So thinner galls have higher parasitism rates and higher number of parasite species as well. So gall shape and size is really important for natural enemy interactions. We found actually two modules, which mean there are two kind of separate networks within this network. Sorry, the topmost galler on the right on the network with Pispondylia brasiliensis. It's a very distinct gall. It's the thickest gall. It's a stem gall with in a rosette shaped, and it has only 4 or 5 different parasitoids, and these parasitoids occur only on this gall and not on any other galls. And they are the parasitoids with the longer ovipositors. So this module is probably driving this pattern, as you can see here. Okay, so when you take a single plant, that's what you find. Oh, here are the galls. Sorry, I almost forgot to show them to you. From A to F, they are increasing in complexity in the structure and also on thickness. So the gall on A, which is from Brugmania elongata, is very thin, so it has lots of parasitoids and a very high parasitism rate, whilst in a kind of a gradient until the F gall there, which is Pispondylia brasiliensis, which is up in the network, as I just told you, is the gall with the fewest number of parasitoids and the lowest level of parasitism as well. So for the next one, the double host plant network, we have 2 recently diverged host plants. They're very closely related. They are actually quite difficult to tell sometimes in the field. We many times had to go to the botany department to check with the taxonomist. There are Mycenia laevigata and Mycenia glomerata, which are 2 Asteraceae, very common. They're climbing vines in the forest you find around here. And as you can see, there's lots of different galls there as well. And we found, as I told you before, we found some of these galls occurring on both species. Some of these galls are occurring only on Mycenia laevigata, and some galls only on glomerata. So we used 3 clustering factors. We combined these different species according to gall structure, from simple galls to very complex galls, from the gallar phylogenetic proximity, because they belong to different genera, and according to host plant identity as well. So from 4,629 galls belonging to 3 monophagus and 4 oligophagous galling Cecidonids, we found 664 parasitoids into 37 morphospecies, which is actually less than the previous example. So here you have the double host plant network. As you can see, the circles are the galling species. I'm sorry, the names for them are in Portuguese, so it's maybe kind of difficult to tell which one is which. And the squares are parasitoids. And this is organized according to modules, so different colors mean different modules, which means a subset of a network, which is relatively separated from the other. And you have in black interactions which are between gallers and Mycenia glomerata, and in red those interactions for Mycenia laevigata, the other plant species. So if we find a higher connectance, 27% of all interactions were found here instead of 21 for the previous network. Modularity was higher here, as you can see. We are using different colors to represent that. It contrasts with all the galloparasitoid systems. these new methods of calculating modularity have not been used extensively for lots of the networks we see in the literature. So it's the right thing to take on too. Parasitoid specificity again was intermediate. I mean, there are specific parasitoids that you can only find attacking one goal, but some of the parasitoids, especially those you can see in the middle of the network, are attacking multiple goals. And we thought that a delayed host tracking hypothesis is probably the best model to explain what is happening here. I mean, because the plants have separated not a very long time ago, there's probably incipient exspeciation, and not every species of the gallers are decided yet between the two species. So some of them can still attack both. Actually, this theory, in theory, this does not take the parasitoids into consideration. We are hoping to highlight that the parasitoids being different between different galls and between different plants could also mean that they are imposing a further selection pressure for these gallers to speciate, to become differentiated, because they having different kinds of pressure from different parasitoids. So top-down effects must be taken into consideration as well. Well, for the final multiple host plant network, as I said, we are comparing active restoration areas with reference areas. We're in this case taking up tritrophic networks because we didn't know how many plants we would find in each area, different from the previous networks. So we include the host plants, gallers, and parasitoids. We still have lots of analysis to do. This is only preliminary. We again found lots of galls, 4,132 in 89 morphotypes and 46 host plant species. We also have 1,175 individual wasps and 62 morpho species, so it's quite large diversity. As I said, very preliminary. We're only considering gall parasitoids for now, only the higher trophic levels. Connectance in this case was really low. It was only 6% of all connections, or all possible connections, were realized, comparing to 27 and 21 as before. And specialization, because of that, also was really high. It was 0.77. You may recall in the first network was 0.55. And we have the galls reported in the literature 2 years ago, and this work really requires further thinking and further analytical solutions. Here you find the tritrophic web. I'm sorry, this is a complete mess. It took me hours yesterday to build this, and it still does not look very good, just because the order of the parasitoids is just it's alphabetical instead of optimized order to reduce this crossing of these lines. As you can see in the bottom, you find the plants. Just above in the middle, you find the gallers, and gallers are attacked by a huge diversity of this parasitoid wasp. It's really hard to make sense of this large network, I know. But so we break them up between native and restoration areas. You can still see, this is just for gallers and parasitoids. You can see that structure. I mean, how many lines cross, how thick these lines are, the diversity of line thickness that you find here. So some parasitoids are very numerous, some are very rare, but it looks the same from comparing, taking a look at the native and the restoration. It looks the same. When you calculate a set of different indexes from the networks, you see connectance is really the same, almost the same. It's 9% for the native, 10% for restoration. Specialization is also, again, very high, 0.8 for both. The number of modules in each of these networks is nearly the same, 5.5 for the native ones, 4.5 for the restoration ones. And robustness for the parasitoid level, which means resistance in terms of extinctions of this parasitoids, is nearly the same for both situations. So which is kind of good. It means that restoration seems to be working. It means that areas that have been restored for the forest, you can already find a very similar set. Not only a very similar set of plants and galls and parasitoids, you also find very the networks, I mean, communities structure in the same way and probably working in the same way between native and restoration areas. So our preliminary conclusions, there's still, as I said before, much to do. Connectance seems to decrease with larger scale or scope of the network. That is to be expected. When you look at one plant or a few plants, you expect more connections because parasitoids would have access to all these goals very easily. The goals are, after all, on the same plants, perhaps not in the same individuals, but on the same species. So parasitoids would probably look for those goals. But when you take up the whole community, let's say, you end up finding very little connections between them because, of course, wasps are not going in from every single plant looking for those goals. Also, because you're taking more samples of a single system, you probably find a deeper view, so you're likely to find more connections. We need to explore this further, as I said. Modularity seems to increase with larger scale scope of the network too. So it means, again, it's the same question: how do parasitoids find or attack galls? They're probably looking for the plants in the first place, so they would not attack galls on different plants. And we end up with few small modules, maybe due to the amount of sampling as well. But there's still so much more to explore in terms not only of the network structure, but also on taxonomy. I mean, there's lots and lots of different wasps. We may be leaving a few species out because we may not be able to separate them adequately. So there's lots more to do, really. So thank you very much for your attention. Thank you to Anna for help during all field work and initial analysis. Thanks to everyone at our lab and to our sponsors. And I'm really glad to answer any questions you have. Thanks a lot for having me.
Thank you very much for your presentation, Milton. So now we are gonna have the last presentation of this session with Jack Schultz. A minute. Hello, Jack. Good afternoon. Your microphone is off, Jack. Sorry.
All right. Now do we have it?
Yeah.
Okay.
Can hear.
Well, I want to thank the organizers for inviting me. I certainly wish that we could be there. We still have a couple of feet of snow on the ground here. But it's been great to actually see the faces of so many investigators whose papers I have read. And I've really been enjoying all the fine work that's being presented here. We're going to talk about genomics a little bit. And I want to start off by pointing out that this is not a leaf. It was going to be a leaf. All of its cells were programmed genetically to become a leaf until a cyanipid wasp came along and reordered the expression of the genes in the leaf cells. The fundamental underlying difference between any gall and its host tissue is which genes are being expressed when and where. After all, the insect has only plant genes to work with, so pretty much all gall formation is a rearrangement of naturally occurring genes. We're seeing a lot of attention given to transcriptomics, the study of gene expression in galls over the last few years. I think there are maybe a dozen papers assessing gene expression in various kinds of galls now. And I thought it would be kind of interesting and maybe even fun to try using the transcriptomic approaches to focus on understanding one little piece of the development of a gall that we work with, the leaf gall formed by the aphid relative Phylloxera. Phylloxera females emerging from overwintering feed on mesophyll from the top surface of the leaf, which induces the formation of a sac-like gall on the bottom of the leaf. And you can see on the right a female, fat and happy female, producing eggs, which will hatch into nymphs that will exit through the top hole and go seek out new leaves in which to get galls going. The very first thing that happens.
I'm sorry to interrupt you, but the audience is asking if you have any presentations to show, or
What?
Presentation is not.
You're not seeing the presentation?
No, it's not here on the.
When I first started, I thought you said it was there. All right, share screen, entire screen. Now tell me if it's there. Is it there?
No, it's still not here.
Oh. Okay, I'm in. Boy, it's showing on my, let's see, try again. Let's try one more time. How about now?
Now it's everything okay.
Yes, you have it?
Yeah.
Oh, okay. So you've heard everything I've said. Now I can just go through the slides. This is not a leaf. It was going to be a leaf, but a cynipid wasp rearranged the expression of the galls into leaf cells. And that's pretty much the underlying basis of all gall development, the changed expression of plant genes from what they were going to do in the original tissue to what they need to do to produce the gall. We're seeing lots of attention to the study of gene expression in galls these days. There are about 12 new papers over the last few years taking that approach. That approach we call transcriptomics, the study of gene expression, in fact, expression of all the genes. And I thought it would be interesting to use that approach to try to understand one tiny portion of the development of the gall that we work with, Phylloxera, which is an aphid relative that forms galls on the underside of grape leaves by feeding on mesophyll from the upper side of the leaf. The very first event that occurs in the development of this gall, and in fact occurs within just a few hours of the females beginning to feed, is the formation of something we call a feeding disc, a circle of, or a disc of about 30 cells across that expands by virtue of cell growth with almost no cell division. You can see in the graph on the left here that the cell density remains well below, in fact decreases continually compared to, in that area of the gall compared with comparable areas of the leaf. So this area of the gall at the very beginning expands until it abuts normal and unchanged leaf material. We'd really like to know how this works. What causes this? What is she doing to get this? Oh no, get this to work. Did I disappear? No. Okay. So we have an hypothesis about this. The hypothesis that we'd like to examine is that auxin is involved in the expansion of the feeding area to form this disc. Why auxin? Well, auxin triggers cell expansion by loosening cell walls. It can move cell to cell, which might be necessary to form this disc. It's concentrated in many galls, as I'm sure most of you know. It's synthesized by insects. In fact, a recent paper suggests that it may be synthesized by almost all insects. And it regulates many other things that happen during gall development. It may, I should say, get some help from a couple of other hormones as well. So what kind of evidence can we seek for auxin's involvement? Well, obviously, we could do analytical chemistry to measure the concentrations of auxin, but we're talking about doing that with very few cells, a very tiny piece of tissue. And while some specialized labs can do that, it's not straightforward. Some have had success tagging auxin with the histochemical techniques. I know Rosie has done that and her colleagues. But we're going to take a different approach. We're going to take the transcriptional evidence approach, in which we ask, are there genes being expressed differently in the gall that provide auxin synthesis or perhaps responses to auxin? So the first thing we have to do, and again, we're going to go through the steps here. First thing we have to do is find genes expressed in the gall and leaf. So the way we do that is we extract messenger RNAs, transcribe that to produce DNA fragments, and then we ask whether or not our DNA fragments fit in any particular portion of the reference genome, which in our case is grape, and there is a published characterized genome for the grape. If our fragments match portions of the grape genome, that suggests that we have found a grape gene sequence, and that we then have identified a grape gene in our samples. Are they different between galls and leaves is the next question. Well, again, we extract messenger RNAs, synthesize DNA fragments, but this time we send them off to a high throughput sequencing facility that returns to us a count of the number of transcripts of each gene. You can think of this as also as counting the number of messenger RNAs. That's pretty much the definition of gene expression. So they've given us the number of transcripts, which is the expression of each grape gene found in the leaf and the gall. So we now have a set of loci, or putative grape genes, and the relative frequency of those genes in our tissues, being expressed in our tissues. And we can compare those two sets of numbers statistically, which gives us a statistically significant statement of the gene expression differences between the gall and the leaf. When we did this in our system, we found 26,346 protein coding genes altogether in the leaf and the gall. That's a very large portion of the grape genome. So we were very effective and efficient in getting, pulling out the genes from the grape genome. 11,049 of those genes whose expression had expressions that differed between the gall and the leaf during at least one gall developmental stage. So those are the genes that presumably are actively engaged in forming the gall and distinguish the gall from the host tissue. Now, I'm using the word gene loosely here because all we're talking about are nucleotide sequences in the grape genome that conform to the definition of a gene, but we have no idea what they are. We don't know what proteins they encode, and without that, we can't tell what they're doing. So the next thing we have to do is identify the proteins that these unannotated grape sequences encode. And the way we do that is to, again, translate gene sequences from the grape genome, for which we have data, translate them to protein sequences, and match them against protein databases to see if we have good matches that tell us what our proteins might be. The obvious choice for this might be a collection of grape proteins. The problem is that too few grape proteins are known and characterized, and so it's not a very profitable way to go. We can do the same thing and have done the same thing with all plant proteins, but there too, what you get is an enormous number of unknown proteins, and that's not terribly helpful either. So we found that the way to go was to see how our proteins match to the Arabidopsis proteome. It has the advantage that virtually, not all, but the great majority of proteins and the genes that encode them are well studied in Arabidopsis. And so we can sort of depend on the knowledge base that's been developed there. We make those matches in the way you would expect, and that is a direct comparison of Arabidopsis protein sequences with the sequences that we have from our grape genes, answering the question, what Arabidopsis protein sequence is most similar to each of our grapevine sequences? That gives us a matched set of loci. We now have a set of putative grape genes that are matched to very similar, very closely related Arabidopsis thaliana genes. In fact, they're called orthologs, based on their protein structure. The really nice thing about this is that there's lots of information about the Arabidopsis genes and their proteins available to us. In particular, we can use the Arabidopsis Information Resource, or TARE, to understand what we may have in among our grape genes. So we can take an Arabidopsis gene number like this one and look it up in the Arabidopsis Information Resource. we find it and we discover that it tells us that that gene encodes a protein called yucca-6. It's a flavin monooxygenase, and it's involved in auxin biosynthesis. This kind of information, I can't emphasize enough, is extremely hard to come by for other plant species. I mean, it is coming. Everybody is working hard to gather this kind of information, but it's still too scanty to make much use of. So in addition to now having possible identity, we have the numbers we got from the high throughput sequencing people that tells us how many copies were present. And that can be, the differences between the gall and the leaf tissue can be expressed as the base 2 logarithm of the number of transcripts in the gall divided by the number of transcripts in the leaf for each gene. And that gives us a relative difference between the gall and the leaf. So what we see here is that in gall stage 1, the one we're talking about, this gene that we looked up, that's yucca-6, this auxin synthesis gene, is 16 times more active in the earliest gall than in the leaf. And you can see it goes up from there. Well, that's only one of the 11,000 genes we found. And while I spend all of my time doing this, I don't have enough time left in my life to go through the 11,000 genes one at a time. So we can focus our search using a collection of information called gene ontologies. A gene ontology, or GO, is a collection of genes grouped together on the basis of their biological function or their molecular activity or their location in cells. You can ask any of those 3 questions of the data. Again, TER, but other locations as well, has collated these gene ontologies. And we see that, where'd we go? Okay. Ontology 00009850 is a collection of genes involved in auxin metabolism. It's called auxin metabolic process, and it has at least 70 genes in it. So we can ask, are any of these 70 genes being expressed in our galls or our leaves? And the answer is yes. By matching the sequences of the genes in that collection with ours, the 11,000, we find that we had 46 matches. That means that 46 of the 70 genes in that GO category were differentially expressed between the gall and the leaf. Now, I should point out that we're really interested in the earliest 2 gall stages here, because that's where this expanding disc develops. And here we have the list of genes that were expressed more in the gall than the leaf, and then some were downregulated in the gall compared to the leaf. In those two stages, 19 of the 46 genes were differentially expressed, 11 upregulated, 8 downregulated. But then we have a problem. What does it mean to have these genes differentially expressed in terms of what auxin is doing? I mean, if you study all 4 gall stages, there are 46 genes contributing to some kind of auxin output. So what does it mean for auxin concentrations or activity to have this particular pattern of expression of these particular genes? Well, here we enter what I call the literature rabbit hole. You really cannot answer the question, what is the summation or the outcome for all of these 46 genes in terms of whether auxin is present, whether it's being synthesized, what it's doing, unless you know what each one of these genes does. And one easy reason to see for that is that some of these genes suppress auxin production and some of them promote it. Gretchen Hagen 3.6 here, which is downregulated in our early gall, deactivates auxin. Because it's being suppressed in the gall, that means that it's contributing more auxin to the situation, despite the fact that it was downregulated. So after we have spent lots and lots of time in the literature studying what these genes do, here's the outcome. Among the genes that were expressed more in the gall than the leaf, there are 11 of them. 9 of them would contribute more auxin to the cells expressed as they are. You already have seen yucca 6. All of these other genes that were highly expressed in the gall compared to the leaf participate in the production of auxin. These two, the blue ones, do not, and they are deactivating genes. So they are presumably reducing the concentration of free auxin. However, they only activate when auxin is present. They respond to auxin by increasing their activity. So the mere fact that they are being expressed here means that they have sensed auxin and are trying to deactivate some of it. If we look at the downregulated genes, we find that based on how they are expressed and what they do, at least 3 of those genes would increase the concentrations of auxin in the cells. 1 could conceivably contribute to making less auxin, and 4 of them would have no net impact on auxin concentrations based on what their activity is and how they were expressed. So the conclusion here, we're beginning to get an answer to our question, is it seems that auxin is being synthesized in the early gall stages. If we did the biochemistry here, we should see increased concentrations of auxin. What about auxin activity? Well, there's another geo category for response to auxin, and that's a list of genes whose expression is upward regulated, or regulated, I should say, by the presence of auxin. We found 26 of those upregulated in the gall, but the story here is complicated by the fact that some of these genes are also regulated by other hormones, particularly ethylene, ABA, and jasmonic acid. So we had to go back into the literature and our own data to determine whether or not these were likely to be the reason for the upregulation of these genes in our gall. And the answer was no for abscisic acid and jasmonic acid. It turns out that the insect suppresses both abscisic acid signaling and JA production and signaling right from the very beginning of gall development. And so they are not available to influence the expression of these genes, and we can assume that those genes were upregulated by auxin. We still can't account for ethylene, which is also produced, and it may be contributing to the expression of some of these genes. Conclusion here is that auxin must be present and is upregulating responsive genes in the early gall. So what do we conclude from all this? Well, we found evidence that auxin is being produced and genes are responding to it during the earliest phylloxera gall stages. And that's consistent with the idea that auxin directs the cell expansion that we observe in the earliest galls. It, however, does not demonstrate it. This is strictly correlative data, and the data are consistent with our hypothesis. But the only way we could really test that hypothesis is by changing the concentrations or activity of auxin, which would require either using CRISPR or transgenic techniques in the grapevine. These results suggest that the plant, since the plant is making auxin in these early stages, the insect doesn't have to supply it. Instead, it would appear that the insect stimulates the production of auxin rather than injecting it. Something I haven't showed you for lack of time is that all of those yellow auxin biosynthesis genes I showed you upregulated in early galls are activated by cytokinins. At the same time, we have found no evidence of cytokinin synthesis in the early galls. So I'll leave it to you to guess what might be going on there. There are lots of cautions associated with this approach. Your host plant needs to have a sequenced genome, and it would be helpful if the genome is annotated. That is, we know what the proteins are and what they do. But it's gotten very easy to develop de novo sequences of any plant species, and so this problem is going away very quickly. It's not always true that similar genes do the same thing in unrelated plant species, and so, for example, some of the genes that we find in Arabidopsis may have been duplicated or refunctionalized in grape. So it is a little dangerous to be sure that the grape, in this case, the grape version of the gene does exactly what the Arabidopsis version does. Many developmental events are not transcriptionally regulated. A lot of them are post-transcriptional, things like protein-protein interactions, degradation, pH regulation. And transcriptomics are strictly correlative. If you want causality, you have to do an experimental manipulation, which may require genetic transformation of the plant. No gene ontology list is ever complete. Genes are added or subtracted every day, and new genes get characterized all the time. So you have to be in the literature constantly searching to find all the genes that are in a go category or may be related to it but not put there. For example, we're arguing that auxin regulates cell expansion, and it does that by increasing the activity of several important proteins, among them expansins, cellulases, and so forth. The genes encoding these do not occur in that GO category that I used to find evidence of auxin activity. Now, there may be a reason for this, which is that auxin creates an acidic environment, and some of these, expansins in particular, are regulated by pH. But I'm pretty sure that auxin regulates the activity of pectin mesylesterases, for example, and that really should be in the auxin response category. So constant literature searching is required to make sense of the data as new functions are discovered. And plant development and hormone networks interact in really complex ways. Most genes participate in multiple pathways and developmental events, as you saw with the auxin, jasmonate, and abscisic acid example. So I picked a really straightforward, relatively simple event to go after with this, but even this one is more complicated than I've suggested here. And then finally, gall development. Really, all this really should be done at the cell and tissue level. Most investigators are sampling entire galls. The activity of auxin that I've been describing here is probably only occurring in a collection of maybe 100 cells. When you grind up the whole gall and you measure auxin, you see no evidence of the auxin activity because it's diluted by the rest of the gall tissue. I am aware of one study only that has analyzed gene expression in different parts of a gall, and the results were profound. The different parts of the gall are doing really different things at the same time. So we really need to find ways to do these studies at a much smaller scale. So I want to thank my collaborators, Dr. Melanie Bodie, who's now at Michigan State University. Patrick Edger is our informatics expert. And Heidi Apple, who is my wife, and I have collaborated for maybe 30 years on these things, and I'm grateful for that. All of this work was supported by the U.S. National Science Foundation, and I really appreciate that. And that's it. Thank you.
Hello. So, thank you everyone for the amazing presentations. Now we will proceed to the questions. A lot of questions. The first question is from Bruno. He asks Kevin. Let's see. Here. Okay. Incredible. There are many mysteries in your Gauss system, such as those bacteria associated with the wasps that we may ignore. Do you believe that they should often occur in other Gauss systems?
Well, Bruno, I'm happy to say yes, they do. That's pretty well known, especially in Cynipidae. I think Graham Stone is attending virtually. He might have some more insight in that. But Wolbachia is very common. It's not to say it's having an influence on the host in all cases, but that's often the case.
I think Graham just published a review on that.
The next question is from Milton. From René, sorry. He asks Milton, systems in which parasitoids are not strong selective pressures, what factors might act as selective pressures driving Gauss speciation? What ecological variables should we pay attention to?
Thanks, René, for the question. It's really a tough one. I'm actually looking at systems with lots of parasitoids, so I'm imagining parasitoids have a lot to do with gall speciation as well. Otherwise, we wouldn't see so many defensive traits in those galls. I would say if you ignore the parasitoids and the predators, then we will end up with the plants. So plants will be the real thing to look for. An example would be the system I talked about, Mecania, which are two plants that are supposedly separating in the process of speciation, and galls are having to choose between them in a certain way, which would probably lead to much more species, to specific species to each one of these plants. So I would guess would be the best answer would be to look at the process of evolution of the plants, of the host plants themselves.
The next question is from René too. He asks Jack, I'm sorry, maybe I missed it. Did you sample along gall development? If so, during early development, how often did you sample the galls? You say that disc emerges in a matter of hours.
Yeah, there were actually two sampling points in that study. One is about 12 hours after the insect begins to feed, and the next one is about 24 hours. We'd really like to do shorter periods, but we just haven't been able to do that yet.
The next is from Rosie. She asks Kevin, how can we start checking the presence of these invisible symbionts?
So the easiest way is with PCR. So just extract the DNA from your insect, and you can test for the presence of Wolbachia using Wolbachia-specific genetic primers. So they will just amplify Wolbachia DNA if it's present in the insect.
The next is from Bruno. He asks Newton, in the system with more parasitoids, did you observe a tendency in more defensive structures on gall surfaces?
I wouldn't say that. Actually, we have found lots of parasitoids in all of the three systems we look at. And in every one of them, we had small galls, not really well protected. And we had thick galls, very well protected ones. We think there's a correlation. I mean, less defended galls will have more parasitoids, but I don't think that's actually what we see. I mean, there are parasitoids for everyone, I guess.
The next question is from Rosie. She asks Jack, I love this information of auxin synthesis genes, 16 times more active in galls than in leaves. In fact, it has been easier to histochemically and immunocytochemically detect auxins in gall tissues than in meristems, for instance. Do you assume it is a matter of higher metabolism activity due to gall's fast life cycle?
No, I don't think so. And I hope you noticed that I used one of your histochemical slides to illustrate the fact that you can do what you described. No, I don't think so. In fact, the regulation of auxin by those genes is generally not tied to overall metabolic activity. So I think it's just I think something has turned on those auxin genes, and my bet is it's cytokinins, but we haven't got there yet.
Okay. The next is from Dennis, and he asks Kevin, there is a relation between Wolbachia infection and host phenological behavior, leading to the univoltism and paternogeny together. It's a question.
You know, Dennis, that's an excellent question. I'm glad you asked it because my wife, Rose, my partner on the project, she's a botanist as well as an entomologist. And we discussed this before, and biotype 2 seems to be specific for Pilostilla officinarum, one type of hawkweed. And in comparison to other hawkweed species, officinarum has continuous growth throughout the season, more so than any other hawkweed. So there's always the availability of tissue susceptible to gall formation. And by somehow making the transition to multivoltinism, it can take advantage of being able to produce multiple generations of galls, but only on officinarum, because the other potential host plant species in the same genus, they don't have that abundance of tissue throughout the growth season available to form galls. I hope that makes sense.
Okay. The next question is from Jose to Milton. Milton, how do you get to the determination of the trophic levels in these systems?
Another good and another tough question. We actually check the galls. That's easily done when we have a single plant, for example, as in my first example. You just open lots of the galls and check if you can find the parasitoids inside. And given the variety of different parasitoids you find, then you check with the number of adult parasitoids you obtain from raising the galls in the lab. So it has checked out for this system as well, but we may be considering there's a chance we are considering some species we could be hyperparasitoids as normal parasitoids. We're not absolutely sure about that. We cannot be sure about that unless we do a very, very deep, very long work for disentangling this complex, potentially more complex interactions, I say. So I'm confident we are showing what's really happening, but it's not 100% that there may be a couple of wasps that are actually in the fourth trophic level, and not on the third trophic level.
Well, the next is from Pedro. He asks, Jack, could the cytokinins come from the insect activities? Salivary substance, maybe?
Absolutely. The insects we're working with are about 1 millimeter in length. And it takes about 1,500 of them to be able to get any kind of decent chemical sampling. And we haven't been terribly successful there. So we're working from the plant back to the insect. And, the fact that we can't find any evidence of cytokinin synthesis, and yet there's a long list of genes, including the auxin genes, responding to cytokinin in the early gall stages, makes me suspect that you are right, and the insect provides cytokinins. All organisms make cytokinins. All insects definitely make cytokinins, and the injection of cytokinins or drooling of cytokinins as insects feed has turned out to be very, very common. So I suspect that that's what's going on too.
The next is from Dennis. He asks Milton, would both host and non-host plant species diversity in a small scale help to drive galling organisms and parasitoid speciation?
Yeah, well, that would require probably a whole hour speaking about it. I think at a small scale, we may be able to look at ecological questions much better than evolutionary questions. So I'm absolutely sure about this. The diversity of plants we find, either host or non-host, probably affect the ability of the wasps to in finding these different goals. So that is one question. How would that drive wasp speciation is another question. It certainly drives their ability to find these goals, and they're certainly not finding goals on different plants. So what our network reveals is probably that. But what does it mean exactly? It just means that it's difficult to find your own the goal you want. I would refrain from going further into complex evolutionary patterns. I don't think we have enough data to actually have a good answer for that. I'm sorry.
Okay, the next is from, don't have a name, but for Kevin. In those gall hasps, we have other bacteria whose holes are unknown, Cardinium, and you can read on the screen. Did you find symbionts in addition to Wolbachia?
Thanks for that question. I think that's from Graham. So in this particular species of wasp, what we find is entirely Wolbachia, but in other species of insects that I study, such as spotted ringed Drosophila or capra beetle, we do find things like Arsenophonus and Spiroplasma in addition to Wolbachia. So it seems to be very specific to the host species as to the complex of bacterial symbionts that you might find.
Okay. The next is for Jack from Sarah. Have you used other clustering methods in addition to GEO categories?
Oh, hi, Sarah. Well, we've done some clustering, but it hasn't been all that helpful. The problem is, I made it look as though we can just smoothly go to other people's collections of genes and learn everything we know about, or find all the genes that we want to know about. It turns out that it's not that clean, and I literally have to spend hours in the literature finding, if I can put it this way, finding the genes that are not clustered appropriately. I mean, it's a Wild West frontier situation. Things are coming together, but coming together awfully slowly, even for Arabidopsis. I showed an example of 70 genes in the auxin metabolism GO. I think there may be twice that many that should be there. And it takes a long time to find them. So we've done some clustering, but it hasn't helped solve this problem, which is kind of a literature issue, I think. Good to hear from you, Sarah.
And this is from Nicholas. He asked for Milton. Do you remember which percentage of galls emerged parasitoids?
Hi, Nicholas. Thanks for the question. Actually, there were no. I don't, because we have percentages for each of the galls, but I wouldn't remember. They fall in very different categories. From some galls, we have only 10% emerging parasitoids. For others, we have something next to 50% or even 60%. So it varies a lot. It varies from galling species to galling species, that's for sure.
The next is for Jack from Dennis. Thanks for presentation, Jack. I am happy to learn with you about transcriptome analysis in front to higher diversity in the neotropics. how far we can get without host genome sequence?
Well, I hasten to say that my part of what we're doing is largely tracking down the genes and figuring out what they do and what they are. Our collaborator there, Patrick Edger, tells me that developing a new de novo sequence for a plant that hasn't been sequenced before is a master's level project. And maybe less than that for his students anyway. So I think it's less and less of a problem. You know, I keep looking at, you have a couple of super hosts in the tropics, and you have some timber trees, and I'm guessing that some of those may have sequences. I haven't looked for them. But a molecular lab can develop a new sequence, I guess, fairly regularly now. The problem comes, you know, as I suggested, when we were forced to go to Arabidopsis to interpret, the problem comes with knowing what those genes are in the, you know, in the novel plant and trying to figure out how to develop a way of knowing what proteins they encode and what they do. But it's getting there. It's getting, you know, increasingly easy. I mean, I've spent 20 years working in the tropics, and I'd love to be doing this there.
The next is from K. McCormack for Newton. What is phenological variation like in galls and parasitoids in your study system? It's important in temperate systems, but is it high to expect something different in yours?
Cool question. Thanks. Actually, since I'm living in a subtropical location, it's kind of intermediate. I mean, the winter is important here. Winter is it's a period of 2 months, most 3 months, where you find very little insect activity. So you may find galls that they're probably empty, or they have overwintering insects. But we haven't looked at specific phenological patterns. We would need a very specific sampling system for that. But I could tell you that during spring and summer you find more galls and more parasitoids. That's certain.
From Bruno, for Kevin, or for Jack. Sorry, is it possible to look for genes expressed by bacteria in gall induction and morphogenesis? Do you believe that bacteria may intermediate gene expression in insect gall development?
The answer to the first question is yes. As Ken will tell you, bacterial genes are easy to separate from eukaryotic genes, and so you should be able to find them and assess their expression. We eliminate them when we extract our plants. Sure, bacteria could be playing a role here, but people have not been successful in showing that. And I think, again, what's he using as a handle? Graham, I think, just reviewed that literature and found little or no evidence for microbes playing a role in the development of galls. But, you know, there are lots of gallers and lots of microbes, so it may be a little early to draw a conclusion. We'll see. They definitely play a role in other systems. Yeah, I'm sure everyone here is familiar with the leaf miners that drool cytokinins into their leaves as they feed, and the cytokinins are coming from Wolbachia. But finding other systems like that hasn't been that easy.
For Newton, from Fraser. Have you observed any interesting phylogenetic signal in your networks? E.g., do closely related parasitoids attack similar sets of hosts?
Very good question. We haven't looked at that yet. That is because also our degree of detail in terms of taxonomy of the wasps are really stopping us from going further. We can get to genus with really lots of time spent on that, and we cannot find people to go to species level. So we would need some kind of molecular method to extract information directly from DNA from these guys to make any to make a comparison like that makes sense, I guess. But it would be really interesting to do. Would be a way of having a parallel to the functional side, which we are kind of trying to explore more and more about these networks. We have data on the. I only show you data on the function about the first system, but we have it for the other two. We're just processing information and hoping to publish that soon.
Okay. For Jack, the same person. Would you be persuaded to launch a web-based guide to plant gene function for Gao researchers? I absolutely recognize your cautionary message.
Well, Graham, so far, at least 3 transcriptome studies I know of have come up with more than 10,000 differentially expressed genes. I don't know. You know, I'm old and retired. I don't know how long I have to do this, and it's a lot of genes and a lot of issues. I mean, one problem we have here is that gall development is a a plant development problem. I mean, the plant is doing exactly what it would do to make its own organs, except that it's a novel organ, and the plant developmental biology people aren't invested in this, and they'd be probably much better than I at telling you which genes do what in development. We have to interact more with the Evo Devo and Eco Devo people, I think, on this sort of thing. But I'm not sure exactly what you're meaning, but there's a lot of genes and a lot of functions. I'm glad you see me being cautionary.
And the last one for Jack, were any of the differentially expressed genes in your system unnoted disappears true in other Gauss systems? Is this a failure of analysis or a sign of genuine novelty in Gauss?
Well, I wouldn't call it a failure, but it is a limit on annotation. I don't know. A huge fraction of the human genome hasn't been annotated. It's not easy, but no, it's not a sign of novelty in gallers. In fact, I think we'll find the same genes. In fact, the studies I know find the same genes over and over again. So it's not novelty on the part of galls. Galls are just another organ, I'm sorry to say. But it's the fact that we just haven't gotten around to annotating a lot of the genes in different plants.
Okay, we have a lot of compliments. Thank you for the amazing presentations. Great talks. Many thanks, Kevin, Milton, and Jack. I especially appreciate the comment on the importance of studies on Gall and the tissue and cell levels.
I think we need an integration of the population level, the plant gene level, the plant cell biology level, the microbial level. I mean, events like this bring people together for the first time to talk about levels. I think that's great. And I hope that the cell biology people don't stop doing what they're doing, because it's really important, even as the gene people start to do things.
And I think a lot of compliment, brilliant, thank you, thanks. Every challenge is a Jedi. Thanks. And yes, I think we can go to the video posters. Thank you all.
You're welcome. It's a pleasure.
No, thank you very much. It's a lot of fun for me.
Mm-hmm.
Thanks for the opportunity. I appreciate it.
Great discussion.
So now we will have a few minutes break and we will come back with the video posters presentation. Today with the team Gauss distribution and co-science. See you soon.
everyone, welcome back. My name is Ana. I'm a biological sciences undergraduate student at the Federal University of Minas Gerais in Belo Horizonte, Brazil. We will start with the video posts and presentations. Let me introduce you to Dylan Jones. He's a PhD student at the State University of New York at Binghamton, New York, USA. And Elaine Cupim Costa, also a PhD student in plant biology at the Federal University of Minas Gerais in Belo Horizonte, Brazil. Welcome, guys.
Thank you.
Thank you, Ana.
Okay, if you have any questions, leave them on the chat and they will be answered after the presentations. Let's start.
Hello, everyone. My name is Dylan Jones, and I'm going to talk about how latitudinal gradients and species diversity provides open niche opportunities for poleward range expansions. So latitudinal diversity gradient is one of the most widely recognized patterns in biological systems, with the greatest species diversity usually occurring closer to the equator and also at lower elevations. However, climate change is causing shifts in global temperatures and seasonality, and one response to this is that species will shift their ranges poleward and to higher elevations. However, species are not likely to do this together in a community due to physiological and dispersal limitations. So I study a Gary Oak gall wasp study system with our Gary Oak ranging from Northern California up to Vancouver Island here in green. Our focal gall wasp is the jumping gall wasp, Neroturtus saltatorius, which ranges natively from Northern California up to the Puget Sound of Washington. However, in the 1980s, it expanded its range onto Vancouver Island. In the native range, this is generally what we see, a handful of galls occurring on a relatively healthy looking leaf. However, in the expanded range, we can find hundreds of galls on a single leaf across the island in consecutive years. This has the possibility to negatively impact tree health, which could cascade all the way through this ecosystem. We believe this is occurring due to open niche opportunities in the northern range due to a release from competition. So we believe there is greater species diversity on the mainland in the native range of our focal species, which leads to strong competition, and a loss of species diversity in our gall formers on the island, which leads to weak competition and ecological release. So in 2019, we surveyed across 6 regions, R1 being here in Northern California, and we go up in latitude to R6 on the island. In each region, we have 3 sites with a total of 540 trees across 3 surveying periods. We collected 25 syniptic morphotype that we place into 3 gall group types. Here in blue are our detachable leaf gall formers, where we'll find our focal species. Additionally, we have our integral leaf gall formers in purple, and then stem gall formers in orange. So the first thing we looked at was our community composition, which is looking at the presence and absence of species across regions. So our galls are color-coded as previously seen, with our focal species abbreviated and in black here. Regions are also color-coded, and this corresponds to where they fall in latitude. Dots are sites, ellipses are our regions. So generally what we see going from R1 to R4 is there's changes in community composition with some overlap. However, community composition is distinctly different and separated from the rest of the regions on the island. We believe this is in part due to 2 factors. One, an absence of synipid detachable gall diversity on the island, and higher abundance of our focal species on the island. With this, we do see latitudinal trends for our synipid composition. Additionally, for synipid richness, we see a decrease as we move up in latitude with the fewest number of species occurring on the island. For abundance, excluding our focal species, we see a reduction as we move up in latitude. However, our focal species has a positive relationship with latitude, having its highest abundance in the expanded range. Lastly, what we wanted to do to see if species were associated with one another, so we did partial correlation networks, which is partial correlation analyses within region for our 6 regions to see if gall formers were associated on trees. We have our correlation strengths here, and if it's significant, it's a solid line. These are the lines that connect our gall formers, which are these dots here. Gulf farmers are color-coded as previously seen, with our focal species here in off-white, which you will find here. So we know that species diversity decreases as we move up in latitude, going from R1 to R6. However, we also see species association with our focal species, specifically detachable Gulf farmers, are completely absent in the expanded range, with no associations in the expanded range. So with this, we do see latitudinal diversity gradient does affect synapid diversity for our regional species pools. So as we move up in latitude, we see changes in richness and synapid composition. Additionally, we believe that this provides smaller species pools in the northern ranges, leading to open niche opportunities through a loss of associated species and competition. With this, I would like to thank my advisor for all their guidance throughout this project, field and lab help that we received, the landowners for allowing us to survey on this land, and indigenous tribes who historically owned and lived on these properties and should be recognized for this. And lastly, Binghamton University, NSF, and National Geographic Society for all their funding. Thank you.
Hi, I am Elaine. I am doctorate student in plant biology at Federal University of Minas Gerais, and I will talk about phenological synchronism strategies and abundance of 5 Lopesia galls, Cecidology, with the superhost Mimosa gemulata fabaceae. The study was done by me, Elaine Costa, and by collaboration with Vitor Martini, Aline Santos, José Pires Lemos, and my 2 advisors, Denis Oliveira and Rosi Isaías. The Mimosa gemulata hosts 4 bivalve-shaped galls: the lenticular, the granuloceolate, the brown lanceolate, and the globoid. The 5 gall morphotype is the clavate galls. The 5 galls are induced by 5 undescribed Lopesia species, and the 5 galls share the same induction microsite, the Mimosa gemulata pinna-hax. We use phenological analysis of the Mimosa gemulata and of the gall life cycles to evaluate the phenological synchronism strategies among galling insects, sharing the same microhabitat in a single superhost plant. In addition, we related the abundance of the 5 Lopesia galls with the plant phenophases, as well as the neotropical savanna climate. The vegetative phenophases of the Mimosa gemulata are leaf flushing, mature leaves, and leaf falling. The reproductive phenophases of the Mimosa gemulata are inflorescence buds, inflorescence anthesis, immature and mature fruits. The gall life cycles were considered the gall phase, the induction, the growth and development, the maturation, and the senescence. The climate of the region has marked seasonality. The rain season ranges from November to April, and dry season ranges from May to October. Mimosa gemulata is a semi-deciduous shrub with periods of leaf flushing and leaf falling and reproductive phenophases related to the constrained periods of drought and raining in the neotropical savanna climate. The generation of the 5-leaf gall moth species on Mimosa gemulata revealed multivoltine life cycles for the 5 Lopezia species, which may have 6, 4, or 3 generations in the years, as illustrated here. The success of the Lopezia species depends on the synchrony on the induction phase in black lines with the leaf flash in green lines, milling in the rainy season. The other important strategy is the asynchrony of the life cycle of Lopezia species, which is favored the no overlapping of the gall induction periods. The graphics demonstrate the abundance of the 5 Lopezia galls was higher during the rain season and decreased during the drying season. We see in the canonical analysis of the principal coordinate that the flowering phenophase did not affect the life cycle of the 5 Lopezia galls. Differently, the phenological synchrony of leaf falling and fruit phenophases related to a decrease of the population of the gall in Lopezia and consequently the low gall abundance. We conclude that the gall life cycles and abundance of the 5 Lopezia galls follow the phenology of Mimosa gemulata, which obeys the seasonal pattern of the water availability in the neotropical savanna climate. I want to thank everybody for your willingness to listen to me.
Thank you guys for your presentations. We have a question for Dylan here from Jose. Is there any functional implication for gall position, detachable or integral?
I was muted. Yes, so great question. So what we're kind of trying to get at here is thinking about the gall diversity and positionality playing a role in maybe more direct or closer competition. So species that are maybe similar in positionality or similar in gall morphology are likely to maybe compete for similar spaces on the leaf, but also through apparent competition, which is thinking about indirect competition. So species that are similar in morphology or gall positionality might share more parasitoids, so then that could play a role in that. And so when we get to those correlation networks, we think about how other detachable galls are associated with our species. So really kind of trying to associate niches and then building into that kind of competition complex there.
Okay, thank you. Now we have one for Elaine from Gabriela. How can you be sure that there are 4 distinct species?
So I collected the histories of the Lopezia and sent for Professor Valéria Cid Maia, is expert in identification of Cecidology, and see the 5 Lopezia galls because I don't is different or not, and see look at the larvae and family and family adults and see is different because the length of the flagellomerus of the family, and because the prothoracic spatula of the larvae. I understand me?
I think she did. Thank you. We have another one for Eleni from René Carneiro. Which morphotype is the most abundant? There is no synchrony between any of the galls at any time.
So the most abundant morphotype is the brown bivalve-shaped galls, and no synchronized between the induction phase. But I look the all galls all the time. But the induction phase is asynchrony, okay?
Okay, thank you. Another one for Dylan from McCormack. How many years of study do you guys have for populations of galls and their parasitoids? Does it fluctuate widely?
Great question. So for the data I just presented, that was just based off of 2019 data. So we collected off of the developmental period of our focal species, but we also have data for Neuroteris saltatorius, the jumping gall wasp, from 2017, 2007, and 2008. My advisor's been working in the system for a while. I haven't calculated that specifically, but I know at certain sites, the variation of the abundance of our focal species does vary, and I think probably that also occurs for other gall species as well. It's not something I've specifically looked into, but I know that in 2019, some of the sites that we were sampling, that we also sampled in 2017, some of those gall abundances were slightly different at some of those sites as well. Oh, and that also could tie into the parasitoids additionally. So right now, we're still collecting and going through all of our emergence to sort out what the synipid species are and what parasitoids have emerged. So once we get that data, potentially we could compare that with our 2017 data as well.
Okay, thank you. One more for Elaine from Graciele. The photos are beautiful. Is the anatomical analysis of these galls related to the period of development of each morphotype?
Yes, yes, I did the analysis of the anatomical development. It is very different among the galls, and I observe that when the gall have the longest life cycle, occur the more investment in the tissue, as typical nutritive tissue, common stored tissue, mechanical zone.
Okay. One more for Dylan from Jose. Are the inducer already identified?
Yeah, so I refer to them as morphotypes in my presentation because for synipid wasps, a lot of these species actually have an agamic and a gamic generation. So they have two different reproductive modes in a single year. But for the most part, they are already identified. There was a couple instances where two species look very similar. So we, in our analyses thus far, have put those together to be conservative. But again, as we're kind of going through the process of identification, we'll be able to further identify whether they're the same or not. And then futuristically, we're hoping to do some DNA barcoding as well, which will further elicit if they're similar or different species. But for the most part, we're pretty comfortable with the snippet identification. It's more of the parasitoids where we're working towards kind of sifting through the diversity of those right now.
Okay, thank you. Thank you guys for your time. I will now call Pedro Henrique Pereira Gonçalves. He's a master's student in ecology at the Federal University of Uberlândia in Brazil. and Nina de Castro Jorge. She's a PhD student in plant biology at the Federal University of Minas Gerais. And Elaine is already here, so welcome, both of you.
Hello, everyone. Good afternoon.
Hello, everyone.
Let's start then.
Hello everyone, my name is Pedro Henrique and I'm a student from the Graduate Ecology program here at the Federal University of Uberlândia. Today I'm here to tell you a little story about a research that was developed by Denis Coelho de Oliveira and Yara Costa Rezende, and it was illustrated by João Argrelli in the BioHQ comic biology book that it is available for purchase. So let's enjoy the ride. In the Cerrado biome, there are several types of vegetation that interact in different ways with insects. Among these vegetations, the veredas stand out, with soaked soils where buritis and capuchinhas are found. Capuchinha is scientifically called Macaira radula, and it can be recognized by its flowers. In the veredas where Macaira radula lives, there is also a species of moth, Paleomystella oligophaga. It is likely that this insect finds Macaira radula through the chemicals that this plant releases into the environment. Then, the insect lays its eggs on the plant, next to the axillary buds of the branches. Here it is, the insect laying its eggs. After laying, Paleomystella oligophaga leaving the egg in the Macaira radula. When the egg hatches, the moth larva begins to feed, initiating a local response from the plant. The plant cells in the place where the larvae of the moth feed start to grow around the insect. The new structure around the insect that offers shelter and food is called gall, and the Paleomystella oligophaga is called galling insect. In nature, there are other galling insects that induce galls in different plants, galls with different shapes, such as those that occur in leaves of Copaifera luxdorfi. Here it is an example, popularly known as oil stick. The gall induced by Paleomystella oligophaga in Macaira radula has color variation. What would be the ecological function of this? Inside the gall, the larva of Paleomystella oligophaga is protected. As the moth feeds on the gall, we notice an increase in the number of cells. The cell division is more intense inside the gall, next to the larval chamber. We realize this because the cells are smaller and in greater quantity. In the smaller cells around the larval chamber, nutritious compounds accumulate that the moth uses to feed itself during its development. In the outer tissues of the gall, with larger cells, there is an accumulation of phenolic compounds, terpenoids, and alkaloids. These substances would theoretically act as a defense against natural enemies. However, it doesn't seem to work. In the same place where the Macaira radula bushes and the Paleomystella oligophaga moths lives, wasps of the genus Pliny also lives. On galls attacked by wasps, the galling insect dies, and new adult wasps hatch. On galls that have not been attacked by wasps or other natural enemies, the larvae of Paleomystella oligophaga develop, become adult, insects, and hatch, following their curious life cycle. That's it. This is the story that I came here to talk to you about. I hope you all enjoyed. And thank you all for your attention. It is a pleasure to be here. Bye.
The Curious Case of Ants That Live in Galls, Nina de Castro Jorge and Rosemary dos Santos Isaías.
Once upon a time, there was an ant named Nasomirmex, Naso for his close friends. Naso and his family live at Itacolomi Park in Ouro Preto City in Minas Gerais State, Brazil. They are always walking around the Candeal next to the cattle, looking for food and a safe place to live. Naso loves the Candeal. He thinks the candeia trees are very pretty, with their flowers grouped as small white balls, and many trees are all together forming the candeal. Isn't it a spectacular show? One day, Naso, that's very courageous, decided to climb the candeia trees. He has heard that there was a sweet juice named nectar coming from the plants up there, and that many ants like it. And there he goes, looking for the dreamy nectar. But Naso was not prepared to what he would find there, and he was greatly surprised when, in one of the branches, he found a small ball that looked like part of the plant, but that he had never seen before. He tapped with his legs on top of the ball and called.
Hey, I can hear you. Who are you? How did you get inside this ball?
Naso did not expect the answer and got paralyzed when he heard a voice saying,
Hi, I am the Neolasioptera. You can get away from here. You cannot touch me. I am protected inside here.
Naso got astonished and speechless. He did not know what to say, indeed. Where were that voice coming from? And why did it think he would attack? Naso decided to go down the tree and call his friend Camponotus to follow this adventure. Cam was very smart, and Naso knew she should have an explanation for all that madness. Naso and Cam came back to the tree to talk to the inhabitant of the bizarre ball. Cam began the conversation.
Hi, Mr. I don't know, Neolasio. May I call you Neo? Me and my friend are curious about your house. We have never seen something like this before. May you help us understand how did you construct this plant house?
Well, sweetie, aren't you enemies? May I be sure you are not trying to take me out of here?
Of course not, Mr. Neo. We are ants and are very curious about your home. We just want to understand.
Okay, that's all right. I will tell you. This history comes from a long, long time ago. One day, my great-grandparents decided to put their eggs in some plant branches. They thought the eggs would be protected up here. They did not expect the plant to react to the eggs in its branches. And you cannot imagine how hard it was to construct this ball. The plant reacting and my great-grandmother counterattacking, and from this battle comes a ball like this one, where I live today. That's it. Afterwards, when the larva developed,
it could dig a tunnel and escape from inside the ball. It told all the family how it survived inside the ball. And nowadays, all my family lives in balls like this.
Whoa, Mr. Neo, your history is very interesting. But tell me, does your house have a name?
Well, we call it our home, but I have heard some curious people that come here and try to understand how we live, naming our home as goals.
Oh, Gull, love it. It's a nice name. But what happens with the Gull when you leave it?
Unfortunately, it dies. It is our presence inside it that makes the goal alive and growing. When we leave the goal, it stops growing and dies.
Kim, at this moment, had a brilliant idea. She was tired of walking around the Candeal without a house to be her home, and she started thinking what she could do to live inside a goal when Mr. New left it. And she must be fast, for his smart tone of voice, Mr. New would certainly leave the goal soon. Cam knew a very smart guide that lived nearby in other conveyor branches, Mr. Fungo. Mr. Fungo should know what she might do to live in the goal. Mr. Fungo got enchanted with the idea. A protected place with warm climate and available food? It sounds impressive. Just like wise Mr. New stimulated the goal to be alive, which contributed to its own survivorship. The presence of Mr. Fungo and of Cam family performed a similar role. And then, even after Mr. New left, the goal stayed alive, differently from what happened when one of Mr. New's parents went out and there was no one to live in that house.
Scientific curiosities. Do you know that all living beings have a name, which are their scientific names? These names are created by curious people, the scientists, who study, describe, and name each one of the living beings. The characters of our story are real and they have scientific names as well. Let's find out. The scientific name of the ant Niso is Nesomyrmex, and the name of Cam for the scientist is Camponotus. The smart Mr. Neo, the Goller, was described by the scientists as Neolasioptera. And the Candeia, the tree where the whole story goes on, is the Eremanthus erythropappus.
Hello, you guys. I am Rosi Isaías, and together with Elaine Costa, would like to invite you to know our didactic game, Goals from Inside Out.
The objective of the games Goals from Inside Out is to present basic concepts regarding goals, and then goal insect cycles, as well as biotic and abiotic factors that may interfere with the cycles.
The game may be played by 2 or 4 players. In the first case, the players will use 2 boards and 48 cards, and in the second case, they will use 4 boards and 96 cards. I mean, each player has its own board.
For 2 players, each player has his board representing the life cycle of inducer and its host plant, and begin the game by drawing 5 cards from the previous shuffle deck, with the layers between the boards.
To start the game, our player must have one of these two cards, the off position or the go induction card, and put it in the right place on the board.
Now it is the turn of the second player. He uses one of the 5 cards in the hand or exchange 2 cards in the deck because he needs the opposition or the induction cards, and then place it one on the board.
The players may interfere in the life cycle of the other player by using one of the attack cards that present biotic or abiotic factors. In the case you are attacked, you can use your defense cards that are ligaments or rain.
Who is the winner? The winner is the player or inducer who first completes his life cycle with the board full.
We have the opportunity of presenting this game during the exposition Black Professors of UFMG in our Space of Knowledge. That was a really amazing opportunity.
How can you get the game board and cards? You can find them in the Neutral Calgal Group website, where the game can be downloaded in Portuguese, English, and Spanish versions.
Many thanks for your attention. We hope you can enjoy our game.
Here are our email contacts and the Instagram handles of the UFMG Plant Anatomy Laboratory. See you!
Thank you guys for your presentations. There's a lot of love for them in the chat and also a few questions. The first one for Pedro. Do you already use this material in classroom? If yes, tell me your experience.
Hi, Marcelo. I didn't have the experience of using this material in a classroom yet, but it would be very nice. It is very good to show the people how Gall works and how the Gall structure interacts with the environment. But I didn't use it yet. That's it.
Okay, this one from René. Fantastic synthesis. From how many publications did you extract all this information? How difficult was that?
Oh, I just extracted all the information from the comic book that was published, but the system of Macaria hadula and Paleomystella oligophaga is a very interesting system to study. There are some publications with the system, one from Oyara that made in her graduate studies. She showed that how the interaction within the natural enemies interacts with the metabolism of the skulls. And I did some research that are still in progress with the system that shows that the parasitoids has a specific feeding habitat. This pink pliny parasitoid has a feeding habitat that is called entomophytophagy. He eats the whole body of the gall-inducing insect. He eats the whole larvae of the gall-inducing insects. And after that, he eats plant tissues. It is a very rare feeding habitat that can happen. There is some report in some papers in India, but there are a lot of work that we still have to do with the system. It is an awesome system.
Okay, thank you. Next question for Nina from Marcelo Guerra. Who made the drawings?
No, unfortunately not. I doesn't have talent for that. But I have a friend, she has a son, and he draw for me.
Okay. Next one. Where is it? From Hene, also for Nina. Can you talk a little bit about the challenge of translating scientific language into popular language? Is it too difficult?
So I started with a biological course to be teacher in high school. So for me, this link is very special and is a wish for me. Rosie support me in these ideas, in these crazy ideas. So it's cool to do that because it's about me, it's about my wish, it's about my life history. It's difficult because when we pass long time in the scientific ambience environment, so it's difficult to translate for kids the scientific content. But I love it, so it's cool. It's not difficult, it's cool. It's a challenge, but I love it.
Very nice. This one for Pedro and Nina from Marcelo. How is the presentation of these didactic materials? Reading? Did you think to prepare animations about these histories?
When I was preparing the material for you all, I thought just for reading and putting some effects in the presentation to be more clear and good for you to understand. But when I think to present this material for, I don't know, high school or elementary school people, I think it is good to do an animation, but to do this, we have to have some partnership with some people that it is good at the thing, you know. We are just amateurs. Talent for me.
And for me, this presentation is a small part of a book. We write a book in 3 different languages, Portuguese, English, and Spanish. You can find this complete version in the Neotropic Goal Group website. In this moment, we apply this book in a school. We have a friends, teachers, friends, so they apply this material in high school. But the animation is a cool idea. I think about that because it's more easy to communicate. But in the moment, we just use for a book for reading.
All right, thank you. This one for Elaine. Elaine and Jose, in your experience, which is the part of the game that engages the audiences the most while playing it? What surprises the players the most about the life cycle of a galling insect?
I think you start the game with the induction or oviposition cards within head is cool, and the attack it and defense card is very interesting too. And in my opinion, I think the parasites is very cool and life cycle, and the poop is interesting too.
Okay. One more for Elaine from Marcelo. It is a game for a winner. What do you think about the collaborative games? Your microphone is off. Elaine, your microphone is off.
Oh, sorry. Sorry. I think it's possible collaborating the game, but Rosie, help me. Help me in chat, please. The Go group created the Go group created the game. Is it, Nina?
Should I put her here?
We can create.
Yes.
Okay.
Can you help me? Can you help me? I like.
I was trying to say, but I answered Marcelo in the chat of YouTube. That's a great idea, Marcelo. Let's create. You can help us. I love creating games. Okay? It's a very good idea.
Okay, thank you, Rosie. See you soon. We have a question from... No, we have no more questions here. Sorry. Thank you guys for your presentations and your time. Nina, do you want to say something?
Yes, I would like to thank you, Rosie, Renee, Marina, and Bruno, two voices about my book. So thank you.
Anyone else wants to say something? Okay. One more question popped up. How do children react?
I am very excited about children react because I'm very insecure because I think it's a different way to talk about science. So I'm very insecure about that. But I'm very happy about children react because they understand the history and talk about the history and have many curiosities about goals, about relationships and plants and insects. So I'm invited to talk to children at the school. So I'm very excited for that. And it's cool to see that the charity very interesting about the natural world. So the reaction is very, very fun.
Okay, guys. Thank you for your presence, for your time. Now I will call the organizing committee for a final commentary. So, Rosie, Bruno, and René.
And where is
Hello,
Lenny?
good night.
House? Oh, okay. Forget it.
Hello, how are you?
I'm fine.
I'm very happy
Good.
for this day.
Yeah.
Because we had several good presentations. I take some notes, and we are learning several things about goals, because goals with it's a very interdisciplinary subject. So we are step by step understanding the complexity of the goals, and they have been reviewed as very good models to study symbiotic relationships, interactions, and their impacts in the plant productivity. So the ability of the ability of manipulating and the gene expression, the plant development. It's amazing. It's a very interesting for us plant anatomists and the plant physiologists, the ecologists. So I'm very happy. I'm really very happy. Thank you very much for these incredible days. I loved the presentation of the students. We need more students engaged in this cool science, this theme, because science needs to be upgraded. Do you have comments? And then from you?
No, I just think it's awesome that we are here. Even though the world, especially Brazil, is chaotic right now, we've been going through really hard times, and I'm not trying to romanticize this moment here. This demands lots of efforts and contribution from people from all around the world, and sometimes we don't know what's going on with them. And this is, I mean, the effort that they came here and contributed and participated on this rich discussion is, I mean, I get emotional because it means a lot to me. And being a scientist is an act of resistance. And here we have the amazing products, not only papers, but also things that actually will reach our children and teach them how to look at the environment. And, you know, it's awesome. So thank you all for the brilliant participations here today.
Great. I'd like to say that I'm quite happy to meet all these ladies and gentlemen that kindly agreed to collaborate with us. I'm wondering here, I guess I met all of them due to the interactions resulting in calls. Daniel, Burkhardt, Dalva Queiroz, Valéria Maia, Jusso Moreira, Adriano Cavaleri, that we met yesterday, and today, Berin, Tienko, Kevin Float, and Jack Schultz. Remy Carneiro, we keep on working with goals, but we met a little bit before, as well as Bruno Ferreira. But we are all together just because of these plant-insect, plant-animal interactions, plant-plant interactions that result in goals. And I am quite happy to be part of it. Okay, so Valéria and Marcelo Guerra, Denis Oliveira, who are here in backstage, I suppose, I would say thank you very much for believing in this trip that we are doing together. And I hope we will have more opportunities to be together presentially all over the world, wherever the goals get us. Okay, thank you very much. And we'll see you tomorrow.
So do you want to host the presentations for 15 minutes? If someone can open a virtual room in Meet for 15 minutes and to toast.
Did you open?
No, I'm not. But someone can open and put here in the chat.
Can you open a Meet room, Anna? Or I do it here? I can do it.
I can
just an idea. If you don't want.
No, do it. Let's do it. We have the idea. So
okay, let me just end the stream and then we'll go.
Yes. Then we can go offline and discuss our lives.
Yeah. People, the idea is that now we will share a link for a Google Meet, and those who are here and want to be with us there, so we can have more people on the screen for getting a photo, a print screen of the participants. So be our guests. You can bring your glass of wine, of water, cup of coffee, cup of tea. Okay, just okay, I have to. Let's go to Google Meet. Yeah, maybe.
Okay. So I put in the chat of YouTube the link of this meeting. 15 minutes to drink a beer, a whiskey, a coffee, or Water.
Yeah, it's not now. We have a stop.
No, now.
We are over.
That's
Okay.
now.
So,
Okay.
are we?
Okay.
All
Okay.
right.
Okay.
Thank you,
See you
Ana Maria.
Bye
Very
bye.
good.
We are off.
See you tomorrow, guys.
See you tomorrow.
See you.